A big hello to all my cute little champions. I hope every one of you who is watching this video is doing absolutely great, my dear champions. And as you can see, I am so happy. Why, you may ask, or you may not ask why, but let me tell you. It's because we are actually in the verge of completing the marathon series. So we have completed the 1 marker, 2, 3 marker and the 5 marker questions. And with this, I can guarantee you that we are going to score really good marks in biology. That is my surety from my end. So a lot of you ask me in the comment section, ma'am, if we do this, are you sure that we'll get full on full? Are we sure that we'll get good marks? Yes, I'm sure, which is why I'm standing for hours and basically making a video for which I'm not even sure that people are going to watch it or not. So please, my dear children, obviously if someone is putting their efforts into creating something and that too for longer hours, it obviously makes some sense, which is why they are making it. And it's not just about me, but all the other teachers who are helping you out. So please make sure that you are taking something from all those videos. Okay, I agree that some of us might be really boring at times when the videos go really long, but the thing is, obviously the energy level goes down, goes up, goes down, goes up, but you have to be staying consistent because it's your exam, right? So it should be your motto that whatever happens, I am going to complete it. Let me just put it in 2x speed and listen to what they are trying to say. I hope I'm going to take something back from this particular video and they are going to help me. This should be our motto, and with this we are getting ahead in today's most repeated 5 marker questions. So I have taken questions from all the chapters from which you would be getting 5 marks. Okay, so not all the chapters have a 5 marker question in the question paper. So there are 3 blueprints and there are only certain chapters which are going to give you 5 marker. So let's get started, and if you are ready, do let me know in the comment section that you are ready to score full on full in biology. So this was the schedule, my dear children, which we followed. So Wednesday you might have got the model paper 1, and Thursday, as you can see, all 1 marker is completed. Saturday model paper 2 is completed, biology 2 and 3 marker, and then PU2 all important diagrams is being completed. Now biology all 5 markers is scheduled on Monday, that is today, and then Wednesday, not Wednesday, actually there's a quick little change. Tomorrow itself you will be getting the passing package, because we know that your Kannada exam and everything is nearing up, so you do not have much time to study for other subjects, but yes, you're getting the passing package. Whenever you get time, you can watch the videos. Okay, so I hope all this thing is clear. Make sure you are utilizing it, and for those of you who are asking for PDF, my dear children, just put it down in the comment section, or else you do not even need to put it down in the comment section. What am I saying? You just have to go join the WhatsApp channel. In the WhatsApp channel, our team leader has been managing all the PDFs for you. She would be giving it, so you just have to download it. Okay, so there's a link being provided. Do not worry about the PDFs. Yeah, so sexual reproduction and flowering plants was the first chapter of your biology. It will be definitely giving you a 1 5 marker question according to the blueprint. So it carries 11 marks. Out of that, 1 5 marker question will be there. So let's get started. The first question, which is usually asked, picture related to pollination is given below. This is the picture that is given below. Okay, will this pollination confirm fertilization? My dear children, any pollination, what do you mean by pollination? Pollination means transfer of pollen grains from the anther to the stigma. Do you think this process will confirm fertilization? What do you mean by fertilization? Fusion of male and the female gametes. Array, the pollen grains have just come. Here, the pollen grains have to germinate, and the pollen tube has to get inside the ovule. There, the male and the female gamete will be fertilizing. So how can just pollination confirm fertilization, my dear children? No, it cannot confirm fertilization. Okay, pollen germination has to take place. The male gametes have to be carried down to the female gametes. They have to fuse. That's when you get fertilization. So let's see the first answer first. Okay, so no, pollination does not always confirm fertilization. Pollination is a transfer of pollen grains from the anther to the stigma, whereas fertilization occurs only if the pollen successfully germinates, forms a pollen tube and reaches the ovule to fuse with the egg cell. I hope this is clear. Okay, now next thing that we have, what are the floral rewards provided by the plants to the insects to revisit? So you know what, I am a plant. Okay, now there's an insect coming. I have to make sure that the insect, particularly which is going to come over here, will come again as well. Why? Because me being a plant, obviously I cannot transfer my pollen grains like that. So I want this insect to come. When the insect comes and sits on the plant, my dear children, and when they brush throughout the plant in search of nectar and all. That's when the pollen grains which are there on the plant would be sticking on the pollinator, that is the insect. Now this insect will be flying away, going to another flower, and basically unknowingly transfers the pollen grains. So there has to be certain floral rewards for which this insect would be coming. So one is nectar. Apart from that, what do you have? One is nectar. Apart from that, you have pollen grains. Pollen grains are rich source of nutrients, my dear children. So obviously pollen grains are also being carried out. Obviously they won't be eating all the pollen grains. Some might be still stuck on their body, and this can be still transferred. Now shelter, that is also one very important thing. A safe place to lay their eggs. So you know yucca and moth. Yucca is a plant and moth is obviously an insect. Now the moth, when it comes over to the yucca plant, it lays its egg because the yucca is providing a shelter to lay the egg, my dear children. And what does the yucca get from this? Basically, yucca is able to do the pollination with the help of this particular pollinator. So these are the 3 rewards that you should definitely remember in the case of flower pollinated plants. Sorry. I'm so sorry, it is insect pollinated plants. Now, okay, mention the characteristics of flower in this pollination other than the floral rewards. So ma'am, you spoke about the floral rewards, but okay, but let's talk about the other things which are there, which should be necessarily there in an insect pollinated flower. So insect pollinated flowers should be fragrant, it should smell really nice, okay. Apart from that, basically they should be brightly colored. So you know insects cannot see all the colors and stuff like that, but still they have certain sensory receptors in their eyes which can see certain versions of certain colors. They basically not see colors, but yes, they could see something. I am not here to explain that because it might take long, but yes, they can see something, and to make them see something, the flowers which are there should be brightly colored. So flowers are brightly colored and they are fragrant. And now suppose they are small flowers, okay. Now small flowers becomes very difficult for them. So basically what happens, if the small flowers are in a bunch, it will appear big. So basically the small flowers comes together to form inflorescence. Now this is another thing. Apart from that, what are the other features? So see, so insect pollinated flowers are large, they're colorful, they're fragrant, and they're rich in nectar, so that these guys come again and again. Now small flowers, if they are there, it will cluster into inflorescence. I already discussed. Now, animal attraction to flowers. How are they attracted? By color and fragrance. That is, we can leave. Now, flowers pollinated by flies and beetles. That is another thing, my dear children. So, flies and beetles, you know what? They do not like fragrance, good odor. They like bad smell. So, foul odor is being secreted to basically attract the animals. You know, Rafflesia, which is the biggest flower, that secretes something, a very foul odor, and it is just to attract the flies and the beetles and everything over there. Now, apart from that, you should also remember that they would be having a very sticky, okay, they would be having a very sticky pollen grain, my dear children, so that this pollen grains can go and stick on the organism and basically can be moving around. So these are the things that you should definitely remember in the case of insect pollinated flowers, okay. Now I am forgetting one point, so let me go see that and come back again, okay. So yes, I think that's all. There is no point that is being missed. So I thought I should not make you lose any bit of marks, which is why I went and corrected once more. But yes, these are the points that you should definitely write. You don't need to write all this thing because those are not the features, okay. Going ahead to the next question, explain the structure of megasporangium. That means the one that would be producing what? The female gametes, my dear children. So megasporangium in the case of plants is nothing but the ovule. So how do you make it? If you know the structure, you would be able to write it well. So basically, let me tell you the structure. So over here, you have the stigma, the style, which is a part of the female reproductive system, right? Now you have the ovary. We have to make the ovary really big. Okay, now over here, you would be having the ovule, my dear children. Okay, this is ovule and this is the ovary. In the ovarian cavity, there is something that is known as placenta. Now, this placenta is the one that will provide initial nourishment to the ovule. And this connection, okay, which is there between placenta and the ovule, it is known as funicle. And this point where it is connected, it is known as hilum. Fine. Now the ovule says, I am not alone. Basically, I am covered by 2 integuments. So there are 2 layers which protects it. So there is one inner integument and then there is one outer integument. Ma'am, you did not make it completely. Why? Because this end is actually free and there is an opening up over here. And what do you call this? You call this opening as micropyle. And this region is known as micropylar. So I am just writing micropylar region over here. Now, the opposite of micropylar, wherever there is opening, my dear children, the opposite of it will always be known as chalazal end. It will be known as chalazal end. Cool. Now, inside the ovule, what is there? Inside the ovule, there are lots of cells. There are lots of cells which groups together to make something that is known as nucellus. Not nucleus, it is nucellus. Now, one cell of the nucellus, okay, one cell of the nucellus will be the megaspore mother cell, which would be giving rise to the megaspore, and this megaspore would be giving rise to the embryo sac, and this embryo sac will be having the female, gametes. So, this is the usual, formation method. And now, this is the structure of megasporangium. Please do not get confused if the placenta is made somewhere over here and it is connected with the funicle over here. Still, the chalazal end will be at the opposite side. Micropyle will be always the opening, and this would be always the opposite end as well. So, explain the structure of megasporangium. The ovule is a small structure attached to the placenta by means of a stalk that is known as funicle. Okay, the body of the ovule fuses with the funicle in the region that is known as hilum. Okay, the ovule has one or two protective envelope known as integuments, my dear children. These encircle the ovule except at the tip where the small opening, that is micropyle, is organized. Now, the chalaza is present opposite to the micropylar end, representing the basal portion of what? The ovule. Now, integuments enclose a mass of cells with reserved food, and they are known as what? Nucellus, and they provide nutrition to this MMC, okay, megaspore mother cell. Now, nucellus encloses the embryo sac or the female gametophyte. It will enclose the embryo sac or female gametophyte once the cell will be dividing into megaspore, and this megaspore will be developing into what? One embryo sac. So, who will be providing nourishment? Initially, it is nucellus. Okay, usually the nucellus is being used up. In some, they persist as well. And what do you call that? If you know what is this nucellus known as, where the nucellus is still persisting and still provides nutrition, it is found in two plants, that is black pepper and beet. You should tell me the answer in the comment section. Okay, so yes, so that was a question. Now, third question. Picture of a mature angiosperm embryo sac is given below and answer the questions that follows. I told you about the embryo sac. So, basically, my dear children, this is a structure that is formed. What do you call this as structure as? This is embryo sac. And where do you get embryo sac from? Basically, the megaspore mother cell, no, what happens to is that it divides into it undergoes meiosis, and in meiosis you have meiosis 1 and meiosis 2. So, in meiosis 1 it divides into 2, again this cell will be dividing into 2, this cell will also be dividing into 2, that's how you end meiosis. In meiosis 2 it will divide. Now, listen to me, when it started it had 2n number of chromosomes, that is double the amount of DNA that is there in the normal cells, okay, of the plant. But once it has underwent meiosis 1, my dear children, what happens is that it becomes half. So, it was diploid over here, it becomes haploid over here. Now, again it does not get half, it will be still n over here. So, all of the cells which are going to form over here, they are haploid, my dear children. Now, there is a twist in the story. All of them are not megaspores. Okay, all of them, they are not going to form the embryo sac or things like that. 3 of them will degenerate, only 1 will be there, and you will call this as functional megaspore. You will call this cell as functional megaspore. Remember, it is haploid in nature, half the number of chromosomes are there. Now, this haploid cell which is there, it will give rise to a structure like this. Initially, the nucleus which was there in that particular cell will divide into 8. Okay, there are 8 nucleuses being formed, and then slowly cell wall will be forming around each nucleuses that you see over here, my dear children. That's how you get cell. So, now you are asked, which cells or nuclei of the embryo sac produce zygote and primary endosperm nucleus? So, listen to me carefully. What happens over here? So, always remember this thing that you see over here. Okay, this thing which has like finger-like projections over here. You can see over here certain finger-like projections. Okay, this cell will always be synergids. Okay, these cells that you see are synergids. How many of them do you have? There are 2 in number, and they are the best friends of what? What is this particular cell that you see over here? This is the egg cell, the female gamete. The female gamete. So, 1, 2, 3 cells are over here, and this is the micropylar end. So, wherever there is egg cell or the egg apparatus, you call these 3 cells together as egg apparatus. So, this will be micropylar end. Now, opposite of this micropylar end, what do you have? Chalazal end. And in chalazal end, you have 3 cells again. What do you call them? You call them antipodals. You call them antipodal cells. Now, over here in the middle, my dear children, you have 2 nuclei. The 2 nuclei are just laying there. They are not surrounded immediately by the cell wall. So, 2 nuclei are there, and you call this as 2 polar nuclei. But remember, they definitely have a cell wall around it, a bigger cell wall, okay, with lots of space. And now you will call this cell as what? What do you call this as? You call this as central cell. So in embryo sac, how many nucleus are there? 1, 2, 3, 4, 5, 6 and 7, 8 nucleus are there. But how many cells are there? 1, 2, 3, 4, 5, 6, and this whole thing is what? A cell, that is central cell, in which you have 2 polar nuclei. You are asked which cells or nuclei of the embryo sac produce zygote and primary endosperm nucleus. So listen to me carefully. When the pollen comes with 2 male gametes. One male gamete will be fusing with the egg cell. One male gamete fuses with the egg cell. Synergids provide a medium for it to go. Okay, they have filiform apparatus with the help of which it calls like, come pollen tube, come. My friend, your friend is over here. Egg cell is over here, you come and fuse. Okay, so egg cell and one male gamete fuses to form what? The zygote. Cool. Now you are asked about primary endosperm nucleus. So you remember one more male gamete is still there. This male gamete will be fusing with what? The polar nuclei right over here. This male gamete will be fusing with the 2 polar nuclei right over here. So 2 polar nuclei plus 1 egg cell, that is, sorry, not egg cell, the male gamete which is there, it will be forming something that is known as primary endosperm nucleus. So if you are not understanding, I have written over here, listen to me carefully. Okay, the egg cell fuses with 1 sperm nucleus, or basically 1 male gamete, to form the zygote. Now egg cell was haploid, half number of chromosomes was there. The male gamete which is coming, that will also have, the male gamete that is coming, that will also have haploid. Haploid comes together, you get diploid zygote, my dear children. Now to nourish this zygote, you need someone, and that is what? Endosperm. Endosperm. So who makes it? Again, the 2 polar nuclei which was there fuses with another sperm nucleus, that is male gamete, to form the primary endosperm nucleus. It is triploid. Why? Because 2 nuclei are there. Nuclei, both of them will be having half of n number of chromosomes, and now the male gamete also has n plus n plus n is equals to what? Triploid. It becomes into triploid thing. Now the second question is, what is the ploidy of the following? You are asked about antipodal cells. What is the ploidy of this? Antipodal cells were here. I told you all the cells over here, my dear children, will be having haploid number of chromosome. This, all the cells. So what is the ploidy of these 3 cells? They are haploid. They are what? Haploid. You do not have to calculate n plus n plus n. No, you are talking about individual antipodal cells. Now you are asked about primary endosperm nucleus. What is the ploidy of that? I told you right now. N and the male gametes n, triploid. It will be what? Triploid. Now, why the endosperm development precedes the embryo development? What do you mean by that? I told you one cell over here is forming the zygote. The other cells that is coming over here, it is forming the primary endosperm nucleus, which is going to make the primary endosperm. Now, why do you think primary endosperm is formed before the zygote develops into embryo? Because for zygote to develop into embryo, you need to provide nourishment. Who is going to provide that, my dear children? It will be endosperm, which is why this endosperm has to completely form first. That's when you are going to transform the zygote into embryo. So endosperm acts as a nutritive tissue and provides essential nutrients to the developing embryo. Early endosperm formation ensures a steady supply of food for proper embryonic growth and development. So whenever you have scarcity of food, you gather the food and keep it, or do you wait that let the scarcity come and then I will gather the food? You won't have food to gather there. So basically it's just like that. For the zygote to form an embryo, it will already have a reserve thing, and that is endosperm. Done. So this question is done. Let's go to the next question. Explain any 3 outbreeding devices in the flowering plants. What do you mean by outbreeding devices, my dear children? Basically, suppose I am a plant, okay, I am always doing self-pollination. My pollen grains are coming and landing on the stigma, and the same thing is happening again and again and again. This will lead to a condition that is known as inbreeding depression. What do you call it? Inbreeding depression. So even plants also have depression like that, okay. So inbreeding depression, my dear children, where what happens is that whatever you do, probably the pollen grains will still land and it will not, germinate, or it won't be a good result. Okay, it won't be giving rise to a fertile ovule or things like that. So this is inbreeding depression which the plants have. Okay, so basically to not allow inbreeding, you have to look for outbreeding. You want the plants to go pollinate other plants and bring out genetic variations. So what does the plant do for itself? Obviously humans can do a lot for themselves, but how can plant do something for itself? Let's see. So first I will not talk about this, I will be talking about this. That is dichogamy. That is, in some species, pollen release and stigma receptivity are not synchronized. So even though the flowers are there, which has the female and the male reproductive system, still the pollen release and the stigma receptivity is not synchronized over here. Okay, the pollen will be released, but stigma is still not receptive. So obviously, even if it comes and lands, it won't be able to germinate. So this is known as dichogamy, my dear children. Okay, now there is under that you have different terms. One is protandry and the other one is protogyny. So flower in which the anther will mature earlier, okay, than the stigma, you will call this as protoandry. The flower in which the stigma matures earlier, it is known as protogyny. But either way, is pollination happen successfully? No. Pollination will happen successfully, but fertilization is not going to happen over there because the stigma receptivity and the pollen release is not being synchronized at all. And what do you call this as? You call this as dichogamy. Now apart from that, the plant itself thought of something. Let me make it's myself self-sterile. That is, there is self-incompatibility or self-sterility, my dear children. This is a genetic mechanism of the plant. Okay, what is it? This is a genetic mechanism of a plant where the pollen grains do not germinate on the stigma of the same flower due to mutual inhibition. So even they are receptive, still it will not allow germination. So that is a genetic makeup difference that is caused, okay, and it is known as self-sterility or self-incompatibility. Now apart from that, plant thought, okay, let me think of something really new. Let me create unisexual flowers. Unisexual flowers as in there are no male and female gamete right over, reproductive system right over here. In one flower there will be only one, a reproductive unit, either female or male. In that case, obviously you are lessening the chances. You are lessening the chances. For example, you and your siblings are put together in one room. Obviously there are chances that you will talk, you will fight. So what if you are just taken to different rooms and you are kept like that for during studying hours? Obviously you would be studying better. That's not always true, but yes, okay. So yes, so this is the same phenomenon applied over here. So unisexual flowers, if both male and female flowers are present on the same plant such as castor and maize, it prevents autogamy but not gytonogamy. So in unisexual flowers category also you have different problems, my dear children. One is, there are unisexual flowers where the situation is something like this. So there's a flower with a female gamete, was a female reproductive system, and there is another flower with the male reproductive system right over here. Okay, now this type of plants, you call them monoecious plants. What do you call them? Monoecious. Why? Because they have both the sexes in a plant. They have both the sexes in one particular plant. Just that the flowers are unisexual. The flower will be having just one sex. But over here, still the pollen grains can land over here. Still, if you see, my dear children, if you see, the genetic makeup will be still the same, whatever you do. So this is one method, but this is not obviously completely really good thing. So monoecious plants, whatever you do, there are chances that they would not do autogamy. Autogamy is when pollen grains from the same flower lands on the stigma of the same flower. So autogamy is prevented for sure. But what about gytonogamy? Gytonogamy still happens. What do you mean by gytonogamy? Gytonogamy is when there are two flowers on the same plant. If pollination is happening between two flowers, you call it gytonogamy. If it is happening in the same flower, it is known as autogamy. So autogamy is prevented in monoecious unisexual flowers, but gytonogamy is still not prevented. But now if you talk about dioecious plants, my dear children, in dioecious there will be only one sex in one particular plant. Whatever you do, there is only one sex. So obviously you have to depend on another plant. That is the best way. So to produce what? Dioecious plants. So monoecious, as I said, both the sexes will be there on the plant. In monoecious you have two categories. One is unisexual flowers and the other one is bisexual. So either the flowers can be this or the flowers can be like this also. Either way you call it monoecious only. If both the sexes are there on the same plant, you call it monoecious. I am telling you again, monoecious has two categories, unisexual flowers and bisexual flowers. This is an example of bisexual. Before that I told you about unisexual flowers. So if it is bisexual, obviously in this case autogamy will also happen. Okay, so unisexual flowers can be produced, but there is something that is mentioned, that is, if the both male and the female flowers are present on the same plant such as castor and maize, that is monoecious plants, it prevents autogamy but not gytonogamy. Male and flowers are present in different plants, that is dioecious plant, which prevents autogamy and gytonogamy, that is papaya. So this is the best way for outbreeding depression. Okay, sorry, for an outbreeding device. Not outbreeding depression, oh my god. So these are the points that you have to write down. Even if you are not mentioning dichogamy and all, it's fine. Just remember the point. Pollen release and stigma receptivity not synchronized. Self-sterility or self-incompatibility. Production of unisexual flowers. 3 things. Done? Clear? Sorted? Chalo. That chapter is done. Kudos to all my dearest little champions. I am explaining a bit much. Why? Because if someone is there who have not studied the chapter and you're depending on me for explanation, that is why I'm explaining. But obviously I might just speed up a bit more because it might be boring for you and also a bit long. So human reproduction, there will be one 5-marker question for sure. Let's see what are those type of questions. So draw a labeled diagrammatic sectional view of the human female reproductive system. I told you in PU2 most important diagrams. This is one such important diagram, my dear children. make sure you're making the uterus, you're mentioning perimetrium, myometrium and endometrium, the 3 layers. Perimetrium is the outer, okay, endometrium is the innermost, and in between you have myometrium, the muscular layer. Now over here you have the 2 ovary. Ovary will be releasing ovum, my dear children. Who takes it? It is basically the infundibulum. In infundibulum you have finger-like projection which is known as fimbriae, which will be collecting it and transferring it to the next broader region, that is ampulla, and then to a narrower region, that is isthmus. Here you have uterine fundus, my dear children, and the uterus basically it is opening with the help of cervix. So in here there is cervical canal as well, and there is the vagina. Vagina plus cervical canal forms the birth canal. So all this piece of information are really important. Make sure you are drawing it properly. Okay, so this is one important question of a 5-marker question you see. Now next thing, draw a neatly labeled diagram of sectional view of mammary gland. Now this is again very simple, but people tend to confuse it. Okay, why? I'll tell you. So easy way of remembering is understanding the flow. Understanding the flow, my dear children. So please remember always, there are 15 to 20 mammary lobes. First thing that comes into mind should be mammary lobes. How many? 15 to 20. So you can think this is one lobe, this is the second lobe, this is the third lobe. So you have so many lobes in here, and each lobe there will be so many alveoli, the tree-like thing. So this thing that you see over here, okay, they are alveoli. So mammary lobes first, 15 to 20, they will be having so many alveoli. Now alveoli will be opening into tubules, mammary tubules. Now that is not being clearly visible over here, but just understand there are tubules over here. Okay, there are tubules. Now the tubules will be opening into mammary duct. From here it is visible. Here there is mammary duct. Ayo, yeah. So lobe, then alveoli, then tubules are there, make tree branches. Now the tubules will be opening together to form mammary duct. The mammary duct, which is still branch, will be opening together to form ampulla, mammary ampulla. Now the ampulla, which is broad, my dear children, it will be opening into something singular that is known as lactiferous duct. And this is the structure of what? The breast, the female mammary gland. So over here you have the nipple region. This is the areolar tissue that is there. These are the fat. Okay, you have so much fat over here, and in the back side you have ribs. In between the ribs you have muscles. That's all. Okay, so this is the structure of the mammary gland, and you have to remember it. Again, very important question. So this came in this model question paper as well. So make sure you're practicing it and remember the flow. Now draw a neatly labeled sectional view of the male reproductive system. Again, this is important. So from your NCERT, from these 3 questions, one diagram is definitely going to come. So in model question, there are 3 papers, right? In one paper this was given, in the other paper that was given, in the other paper that the other one was given. So make sure you are practicing these 3 diagrams really well, because a question is coming for sure. Okay. Now over here, my dear children, what do you see the diagrammatic sectional view of male reproductive system? It's a very easy thing. Okay, so you have the testis over here. Now testis is basically inside a layer that is known as scrotum. From the testis, okay, what comes out over here? The vast difference is going to carry, the vast difference which is there, my dear children, it is going to carry what? It is going to carry the sperms to the urinary bladder. When it opens to the urinary bladder, it will be receiving a vesicle. So what's it? It will be receiving a secretion from seminal vesicle, my dear children. So I will tell you again, testis is there. From the testis, something is going to come out. What is this? This is vas deferens. Vas deferens is going to open into urinary bladder, my dear children. Okay, but before that, it receives what? A secretion from seminal vesicle, that is the first gland. Now both of them, they come together, they join together, and what is this structure that they are forming? They are forming ejaculatory duct. So it will be having sperm as well as little amount of secretion, and now they are opening into what? The urinary bladders thing, that is what? Urethra. So this is the urethra, onto which ejaculatory duct is going to open. Now we have reached over here, my dear children. We have reached over here. What is happening? Listen, obviously, this is seminal vesicle. There is one more gland that covers the whole thing, and that is prostate gland. It's a singular gland. So seminal vesicle is paired. Okay, seminal vesicle is basically paired, but prostate is not paired. There is only single donut shaped thing in which, through which the urethra will be passing down. And now if you see over here, again, this is the external genital organ, that is the penis. Before the entry of the penis, before the urethra goes to the penis, there is another secretion from bulbourethral gland. From what? Bulbourethral gland. And now the urethra will be opening up over here. This is basically known as urethral meatus. The opening is known as urethral meatus, and this structure over here, there is an enlarged portion that is known as glans penis. It is covered by a skin that is known as foreskin. So you should be remembering all this. That's all. Okay. So at the back side of it, there will be rectum and the anus. So remember, in the case of males, there is only one opening for sperm transport as well as for urine to be passed, and that is urethra. Cool. Done. Chalo. Now, write the schematic representation of spermatogenesis. Write the two events that occurs in the ovary and the uterus during follicular phase of menstrual cycle. So, spermatogenesis is very fun thing. Okay. So, I am going to explain it to you. Listen to me very carefully and then you can see the diagram. So, over here, my dear children, there are spermatogonia. Spermatogonia are the cells that will be giving rise to sperms later on. So, spermatogonia, basically, a few of them divide. Okay. They just increase their number. They want to increase their number. And then one such cell will be there, which is saying that I am definitely going to form sperms, and that cell's name is primary spermatocyte. Primary spermatocyte. How do you get it? Basically, the spermatogonia which is there, it differentiates. It says, it gets matured and says that one spermatogonia, okay? One spermatogonia gets matured and says, I am gonna form sperms. Basically, I am primary spermatocyte. So, how much number of chromosome does it have? It has 2n number of chromosome, that is 46 number of chromosome. Now, this is going to undergo meiosis, my dear children. Meiosis happens in two stages. I hope you know that. So, first stage, it is going to give rise to two cells, both of them. Now, over here, reduction happens. The number of chromosome becomes reduced. So, half of it happens over here. What do you call the cells now? Haploid cells. Okay, and what are the name of these cells? Secondary. This is primary one, so this would be secondary. Always remember, primary has diploid number of chromosomes. So, this is secondary, which has haploid number of chromosome, and this is secondary spermatocyte. Now, again, meiosis happens in two stages. The second stage, it divides, okay, this is meiosis 2, but this cell divides into 2, this cell will also divide into 2. Again, my dear children, will halving of chromosome happen? No, it will be still just haploid. It will be still just haploid. So, over here, what do you call them? So, they are round cells, okay, and sperm, are they round? No, they are elongated. So, these immature cells which are being formed over here, I will call them spermatids. What do I call it? Spermatids, my dear children. They are like kids, okay, small kids. They are not mature now, they are immature. Now, they have to be transformed into something, and that process is known as spermiogenesis. Okay, this whole process is known as spermatogenesis, where the formation of sperm is happening. Now, spermiogenesis basically when these guys are going to transform into what? Spermatozoa. Spermatozoa. Now, they are going to be elongated, bit elongated. Then, from the seminiferous tubules, they are going in the seminiferous tubules, all this thing is happening. Now, they are going to leave the seminiferous tubules, and this is known as spermiation. Okay, aviation, similar like spermiation, as in the sperms are being released from the seminiferous tubules, and probably they that means they are somewhere in the accessory ducts. Okay, they are there in the accessory ducts. Cool. So, this is the thing that you should be knowing. Remember it. So, yes. Spermatogonia, mitosis, they will undergo mitosis, that divide, increase in the number, and they will differentiate from primary spermatocyte. You see one cell over here, my dear children, one cell over here is going to undergo the first meiotic division. Okay, this is the primary spermatocyte, and it is going to undergo the first meiotic division. It is going to form secondary spermatocyte, and then they are going to form spermatids, and then spermatocytes are going to transform into spermatozoa with the help of differentiation. So, you do not have to, like, they have confused it a lot. So, what do? You write this. Okay, you basically write this, and then schematically you can represent in front of primary spermatocyte, you make a cell and then divide into 2 and form secondary spermatocyte, then again divide into 2, you get 4 cells at the end, that is spermatids, and then basically transform into spermatozoa. That's all. Okay. Now, write the two events that occur in the ovary and the uterus during follicular phase of menstrual cycle. So, my dear children, during follicular phase, the follicles which are there, they are going to mature. So, inside the ovary there are follicles. Inside the ovary there are follicles which are known as primary follicles. So, the primary follicles in the ovary grow to become what? A fully matured Griffin follicle. Certain, not all of them, okay, certain of them primary follicles will try to get matured, but only one of them will be allowed to get into what? A fully matured Graafian follicle. So, in the follicular phase, the primary follicles will be developing and maturing into Graafian follicle. You can write that. Now, what happens in the uterus then? The uterus, where the follicles are developing, that means the egg which is there, any time the follicles will be bursting and the egg will be released. So, at this point of time, you have to make sure that the endometrium which is there, it is also reforming, because by chance, if the egg is being released, sperm came, fertilized it, who will provide nourishment? Endometrium has to be there, right? So, basically the endometrium of the uterus slowly regenerates through proliferation, which is why the follicular phase is also known as what? It is also known as proliferative phase, because along with the follicles being generating, the endometrium is also regenerating. This is why it is known as proliferation phase. Done? Cool? Next. Write the role of the following: Sertoli cells, Leydig cells, oxytocin in parturition, menarche and menopause. So listen to me. Sertoli cells, what is the use of it? If you know testis, no. It looks like this. They have so many compartments which is known as testicular lobules. Now if I zoom in testicular lobule, my dear children, what do you have? You have 1 to 3 seminiferous tubules. So this is one seminiferous tubule, suppose. This is second one. And now in between the seminiferous tubules, what do you have? In between the seminiferous tubules, you have certain cells. Okay, and certain other cells as well. Okay, in between, this side also, this side also. So what do you call these cells as? First thing, okay, okay, the red one I'll mark first, just a second. The red one which is there, it is known as Leydig cells. Actually, they do not appear red. I'm just making you to remember, okay. So Leydig cells over here, my dear children, they are going to release androgens. We are going to release the male sex hormones which are together known as androgens. So testosterone, everything comes under this particular category. Now you have white cells also. What are this? These are immunologically competent cells. They are there to fight against any infection and everything is there. So I'm not going to mark it. So that is about Leydig cells which are there in the space between the seminiferous tubules. But what is there inside the seminiferous tubules, ma'am? There are certain cells which are known as Sertoli cells. And you know what? They are also known as nurse cells. And what do the nurse do? It's a lovely profession where they take care of the patients really, really well, right? So similarly over here, the Sertoli cells, my dear children, are going to take care of the spermatogonia. You know spermatogonia, the ones that is going to form the sperms. So obviously you need to provide nourishment to these cells. So who provides nourishment? It is the Sertoli cells. So inside the seminiferous tubules, everything related to sperm happens. That is, spermatogonia will be there, Sertoli cells will be there to provide the nourishment and everything. Outside it, you have Leydig cells which will be providing hormones. Why do you need hormones? For the formation of sperms. From spermatogonia, you need certain hormones. So I hope that is clear. So these two things have to be clear. Okay, Sertoli cells provides nourishment to the spermatogonia. Leydig cells basically produces hormones that is known as androgens, which again help in the process of spermatogenesis. Now talking about oxytocin in parturition. What do you mean by parturition? That means the baby is now going to expel out the body of the female. This process is known as parturition. Okay, now what is the role of oxytocin? Oxytocin is basically released by the maternal pituitary gland. Okay, mother have pituitary gland, and pituitary you have two glands, okay, two lobes, anterior and the posterior. For the posterior side of it, the mother would be releasing something that is known as oxytocin. Again and again and again and again. Why do you think oxytocin is being released? Basically, there are certain receptors in the uterus. Okay, you know myometrium, the muscular layer, in that there are certain receptors for oxytocin. When this oxytocin comes and binds over here, what will happen? The myometrium, which is carrying muscles, my dear children, it will slowly contract. When it contracts, what will happen? The baby which is over here, it will be moving out because someone is contracting right over there. So this is the role of oxytocin in parturition. It helps in inducing contraction in the smooth muscles of myometrium layer. Works. What is menarche? Menarche basically is the first time a female is going to menstruate. Okay, in her menstrual cycle, the whole menstrual cycle, the first time she has her periods, it is known as menarche. Menopause is basically the last time where she have her periods, okay, or menstruation. So how do you write it properly? Menarche, the first menstrual period, signaling the start of the reproductive ability. From this point onwards, she would be reproductively very much able. Now, the end of menstruation marking the cessation of fertility. Write this. It is a cessation, the stopping of fertility, and hormonal changes will be happening in the case of female. So cessation word is really important. Cessation of what? Fertility. From now onwards, the female is not fertile to bring out a baby. Okay, in the similarly, menarche is basically the start of the reproductive ability of the reproductive cycle. You can write like that. Oxytocin stimulates uterine contraction for labor. You can write it properly. That is basically it helps in the contraction of smooth muscles of myometrium. Now, Leydig cells promote testosterone in response to LH. Okay, luteinizing hormone is there. Luteinizing hormone is the one that tells the Leydig cells, hey, you have to release androgens. And the major androgen is what? Testosterone. Now, it promotes sperm production and male secondary sexual characters. Obviously, the hormones which are there, it will be helping in the sperm production, I told you. And also the male sexual reproductive characters, secondary sexual reproductive characters, as in having beard and all the features which are there, okay, enlargement of the penis and everything. Now, Sertoli cells support and nourish the developing sperms. Done. Chalo. I hope children you are understanding. Oh my god, we have completed that chapter also. And now we have reproductive health. So reproductive health chapter carries no 5-marker question. So it is a 6-marker chapter. It will be having 2-marker, 3-marker and 1-marker questions coming off from it. Okay, so wherever I written no 5-marker, that means a 2-marker, 3-marker have come. So the 2-marker, 3-marker videos, you will be finding lots of questions from this particular chapter. So watch it. Okay, now going ahead. Principles of inheritance and variation, a very important chapter, carries 13 marks, and now I am going to discuss 2 5-marker questions out of those. No, I meant there will be 2 5-marker questions coming. Okay, there will be what? 2 5-marker questions coming. So it's not just you zoning out during studies, it's also me who is zoning out during teaching. Okay, so there will be 2 5-marker questions. So you have to make sure this particular chapter is very much clear to you guys, my dear children. If you're missing out, you're missing out on a lot of things. So do not miss this chapter. Now, few autosome linked recessive gene blood diseases occur in human population. Among them, some are related to qualitative and quantitative problem of synthesizing blood proteins. Explain, ma'am, I'm not understanding anything. Easy, listen. So in our body there are 2 types of chromosome. Okay, one are autosomes, which are responsible for all our features like hair color, eye color, nose color, face color, what's the structure, everything. Okay, that is what autosomes who do that. Now we have a pair of sex chromosomes, only a pair of sex chromosomes, that means 2 chromosomes that is there. So how many autosomes do we have? We have 22 autosomes, 22 pair of autosomes, and we have 1 pair, okay, the 23rd pair, 1 pair of sex chromosomes. Now the thing is, whenever you say autosome linked recessive gene disorder, that means in these chromosome, suppose in this chromosome, one particular gene or somewhere, the gene or any other genes, basically they have some kind of mutation, and because of this particular mutation, if someone is having some disease, you will call this as autosomal disease, autosomal genetic disorder, my dear children. Now it is autosome linked recessive gene disorder, that means for each gene you have 2 alleles, my dear children. If both the alleles are affected, that's only when you will be having this particular disease. That is what you call it as recessive gene disorder. It will be only there if both the genes are affected or both the alleles are affected for one particular gene. Now on the other hand, suppose this allele is fine, this allele is mutated, still there is one allele to overshadow its effect and the person will be normal. He or she will be just a carrier. So I suppose you understood what is autosome and what is recessive disorder. Now we are going ahead with the next thing, that is over here, that is among them. Okay, so there are certain autosome disorder where the blood diseases are caused, probably the blood proteins which have to be formed, those are not being produced and things like that. So there are 2 types of diseases, qualitative and quantitative. Okay, so you have to explain the gene disorders which are qualitative and quantitative. I'll give you a heads up, listen to me very carefully. So there are 2 diseases, one is sickle cell anemia and the other one is thalassemia. First thing, you should be knowing how is it caused. Okay, so sickle cell anemia is caused when basically, you know, there is a gene, HBB gene, that is hemoglobin beta globin chain. So in hemoglobin molecule, hemoglobin is the one that carries oxygen and everything, right? In hemoglobin it is made up of 2 things, alpha and beta globin chains. Now for beta globin chain, this is the protein, okay, right? For this to be produced, you have certain genes which will be coding for certain proteins which will be helping in the formation of this. In the case of sickle cell anemia, on chromosome number 11, my dear children, okay, suppose both the HBB genes are mutated. Suppose both the HBB genes are mutated, my dear children, that's when a person will be having sickle cell anemia. Okay, that's when a person will be having sickle cell anemia. Now suppose there is one normal gene. Okay, there is one normal gene, so I am writing, so HBA and HBS. So this is the affected one, the sickle cell anemia affected, and this was the normal allele which is there. This will be overshadowing the effect and still that person will be just a carrier. That person is not going to have the disease, that person will be just a carrier. Now the condition when both the HBB genes are affected, that's when this arises. That's what I was talking about. HbS, HbS. Both the hemoglobin, beta globin chain, hemoglobin, what do you call it? Wait. The gene that codes for beta globin chain of hemoglobin, when that both of the genes are affected, when both the alleles are affected, that's when one thing happens. That is, here, since beta globin is not being produced much, or basically they are producing a defective version of beta globin chain, hence what will happen? The hemoglobin will not be produced properly, which is why the cell that carries hemoglobin will be sickle celled. Will be what? Sickle celled. Just because this is a defective beta globin gene which is produced. Beta globin is still being produced, but what is happening? A defective beta globin chain is being produced, my dear children. Now, when the defective beta globin chain is being produced, the cell which carries this particular defective beta globin chain, it will be sickle celled in shape. Now, it is what? A qualitative disorder, because the quality of the cell is being deteriorated, or the quality of the hemoglobin is being deteriorated, which is why this is known as qualitative disorder. Cool? Here, listen to me again carefully. The beta globin chain is still being produced, but it is defective beta globin chain being produced, which is why the cell that carries it also will be defective. It will be sickle cell in shape, which is why it is a qualitative disorder. The quality of the globin chain is problematic, which is why all the things happens. Now, on the other hand, if we talk about thalassemia, basically in that, what happens? You have alpha and beta globin chains. So, suppose I am telling you there are two genes for alpha globin chain. There are two genes which will be helping in the formation of alpha globin chain, and these two genes obviously will be having their own alleles also, will be having their own alleles as well. Suppose I am saying that this and this is affected, this allele and this allele are affected. Still there are two other alleles which will still producing what? Alpha globin, but the amount will be what? Less. The quantity of the alpha globin which is being produced, it will be less. Suppose 4 of them are not working. So again what will happen? Alpha globin chain is not being produced at all. Okay, the quantity is being reduced, my dear children, if you are noticing it. Now if you are talking about beta globin chain, again, so this is on chromosome number 16, my dear children, where you have the alpha globin chain coding genes. Okay, now on chromosome number 11, just like this, you would be having the HBB, that is hemoglobin beta chain producing genes over here. Still again, over here both alleles, if they are affected, that means there is no beta globin chain. Suppose if only one is affected and the other one is fine, that means there will be production of beta globin, but the amount of it will be what? Less. The amount of it will be less. So if you see over here, the quality is being deteriorated, but here it is the quantity that is being deteriorated. So I will call this as a quantitative. This is a quantitative disorder, whereas this is what? A qualitative disorder. Got it? It arises because of two different type of mutations. So here the mutation is that in the beta globin chain, okay, so the mRNA which is there in the beta globin chain, it should be having GAG, but instead of A, it has U being replaced, which is why there is another amino acid being produced, that is valine. That is valine, my dear children. And if the amino acids are changing, that means the protein which is produced is also going to change, which is why you get a defective beta globin chain. Here there is another reason for this particular mutation. Okay, mutation is happening in the same gene, but it's something else that is causing it. Okay, so in this way you say alpha thalassemia minor or major. Minor is basically when probably two genes are affected, but still two alleles are affected, but still you have something to rely on. Okay, now you have major. Basically, when all the genes are affected, that means alpha is not being produced at all, alpha globin chain is not being produced at all. At that particular case, that person would not be able to survive. That person would be definitely dying off after a certain point of time. Get it? Similarly here also, if both of them are affected, you call it major beta thalassemia. If only one is affected, you call it minor case of beta thalassemia. So this is a quantitative disorder, this is a qualitative disorder. Both of them are happening in autosomes. So chromosome number 11 and chromosome number 16, my dear children. Remember that. Okay, so both of them are autosomes. Only the 23rd pair of chromosome is the X chromosome. So how do you write this answer? I know this is kind of confusing, but yes. So some blood diseases in humans are caused by mutations in autosomal recessive genes, leading to issues with synthesis of blood proteins. These problems can be categorized as qualitative, affecting the structure or function of the protein, or quantitative, affecting the amount of protein that is being produced. Sickle cell anemia, mutation in the HBB gene on chromosome number 11 causes the production of abnormal hemoglobin. It is abnormal hemoglobin HbS being produced. This defective hemoglobin leads to sickle-shaped red blood cells, impairing oxygen transport. Now, it won't be able to transport the oxygen efficiently, which is why it will be causing blockage and things like that. Okay, there is no oxygen transport, there will be blockage because of its shape and things like that, and that's how the human being who has sickle cell anemia would be dying. Okay, here the defect is caused because of the error in the quality of the protein that is being synthesized. The thalassemia is a quantitative problem caused by mutation in genes responsible for hemoglobin synthesis, HbA1, HbA2 for alpha chain, HbB for beta chain. There can be mutation in one allele of a gene or both alleles of a gene, which leads to reduced or no production of specific hemoglobin chains, causing anemia, fatigue. Here the defect arises due to errors in the quantity of the gene which is being produced. I hope now it's clear. Okay, so this question is really important, usually being asked. This is a model question paper question also, so you should be knowing the answer. Now next question, give reasons for the following. A simple cut results in non-stop bleeding in hemophilia affected individual. Now this is again a blood disorder, but what happens over here, not exactly, what happens over here is that a gene that codes for, okay, a gene that codes for a protein that is a protein that is responsible for clotting, you know, usually when we get a cut, blood will be going out for some time. After that it will stop. Why? Because there will be clotting factors which will be coming there and stopping the blood flow. But in this case, a person who has hemophilia will not be producing this particular one particular clotting factor. One particular clotting factor, my dear children, and because it is not producing one particular clotting factor, it won't be able to clot the blood, which is why the blood will keep on going. Now this is a sex-linked recessive gene disorder. Okay, these genes which are there on the sex chromosomes, which is why it is known as sex-linked gene disorders, my dear children. Okay, and this is the case of hemophilia. So usually there are lots of other clotting factors, but even one among them, if it is not there, if it is not working properly, what will happen? Basically, that person would not be able to show the character that is clotting. So how do you write it? Let's see how do you write it. Just a second. Let me bring this thing over here. How can I bring it over here? Yes. Okay. So non-stop bleeding occurs when a single protein that is a part of the cascade of proteins involved in the clotting of blood is affected. So there is a mutation in that particular gene that codes for it, which is why this condition is happening. Now the next thing, Turner syndrome affected females are usually sterile. So Turner syndrome is when, you know, from your parents you get one sex chromosome each. Okay, if you get X and X, it becomes a female. If it is X and Y, it becomes a male. But in the case of Turner syndrome, there is only one X. From the other parent, there is no chromosome that is coming. So you call this condition as XO condition. Now in this condition, what happens? The ovary which is there, it has to be a female, but is not a female now. And now the ovary which is there, it is rudimentary. That means it is underdeveloped. It is what? Underdeveloped. Now if the ovary is not there, will it produce the female gametes? No, my dear children. So obviously this person who is having this disorder will be sterile. So how do you frame the answer again? Females are usually sterile because they have only one X chromosome, leading to underdeveloped ovaries and lack of normal gamete production. So gamete is not there, that's when you become sterile. You will not be able to have kids. Now, the next question. In Morgan's dihybrid cross experiments on Drosophila showed that flies having genes for yellow body and white eyes exhibited less recombination. You know what? Any question that comes from linkage, recombination, things like that, you don't have to burn your head for that. Just look at this. The flies having genes for yellow body and white eyes exhibited less recombination. Less recombination means what? Suppose you have two chromosomes. Okay, you have two chromosomes. I am coloring this chromosome totally up. Now you have two genes over here. These genes, which is one green gene and one purple gene, my dear children. Okay, so usually what happens is that in the case of recombination, okay, when the crossing over the chromosomes comes together, they cross over their substances. So what happens is that, I'll tell you what happens. Part of the chromosome they get mixed match. Suppose this part over here is coming here, this side, and this part is going that side. So the color will be right over here because this part has come over here, and here you'll have the normal thing. But what was here, it will be going that side. It will be going that side. Suppose the green and the purple went that side also. So basically, are they showing enough amount of recombination? Are they promoting new combination? They're still together. They are still together. Are they recombining better? So I'll say their recombination is what? Less. Why, my dear children? Why is their recombination less? Because both of them, they are something that is known as, they are closely linked. They are closely linked, or they are tightly linked. The distance between them is so less, which is why they are like that. Now suppose if one was here, one was here. If one was here, one was here. Let me take that condition also. So purple one was here, the green one was there. Okay, now what is happening? Now when you are exchanging, what will happen? The purple one will go that side, but the green one still remains over here. So this is a new combination of chromosome that you are getting. This is a new combination of chromosome that you are getting. You're totally, entirely changing. First they were all together, but now when you are exchanging, both the genes are in two separate chromosomes. So obviously there is high amount of new combination, recombination. So when the genes are far away from each other, they show more combination, more recombination. When they are close to each other, they show less recombination. So even if you do not know anything, just understand, if less recombination is mentioned, that means they are very close to each other. They are tightly linked genes. So yellow body and white eyed genes in Drosophila show less recombination because they are located close to each other, okay, on the same chromosome, exhibiting genetic linkage. They show linkage. Cool? Works? Understood? Okay. Now, that is basically, recombination is 50%, that means it is the highest form of recombination. Suppose now the question is like, two genes are there and they show 37.3 recombination. What is the condition? So 37.3 is very close to 50, that means they are showing high amount of recombination. That means these genes are far away from each other. Okay, they have too much distance, they are not tightly linked, they are loosely linked. Okay, now they are putting that 1.3 recombination is there, 1.3% recombination is there. That means recombination is less, that means they are close to each other. Okay, works? Now, inheritance of skin color in humans show different phenotypes. Why inheritance of skin color show different phenotypes, my dear children? Because usually what happens is that we have one gene for one particular character. But in the case of skin color, no, the thing is quite different. Suppose there are 3 genes coding for skin color, which is why you and me have different skin tone. Me, my brother will have different skin tone, because why? 3 different genes are participating. This type of inheritance is known as polygenic inheritance. What do you call it? Polygenic, as in different genes are being involved over here, which is why it is known as polygenic inheritance. Now listen to me. Why does it bring out so many changes? So there is an allele capital A, capital B, and capital C. If all the alleles are like this, then the skin color will be very dark. Will be what? Very dark. Suppose capital A is with small a. Now the skin color will be quite lighter than the previous one. Now over here, if again this is also small a, again the skin color will be bit more lighter. Okay. Now here, the skin color is small b, again bit more lighter. Okay. Again here, small b more lighter. Here, small c more lighter. And here also small c, the lightest skin color that you can see. You get it? So because of the alleles which are there, so these alleles are not going to code for that particular melanin or things like that very much, which is why the skin tone becomes less. If there is capital A, that means it is going to produce what? More amount of that particular protein, which is why the skin tone becomes dark. You get it? So that is why it is a dominant thing. This is what recessive. In recessive condition, when the condition is like this, AA, BB, CC, this when the skin color is the lightest. It is what? The lightest. When the condition is like AA, BB, CC, that's when the skin color is the darkest. Now if the condition is like this, you are asked about what is the lightest skin tone genotype. Choose the genotype. This should be the lightest skin tone genotype. Okay, you get it? This is also light, but the lightest version is this one. Cool. Should know about that. And with that, we solve one more question related to this particular topic. So I was discussing one question, but you got the answer for 2. You can remember like that. So inheritance of skin color in humans show different phenotypes. Why? Because It is polygenic inheritance, where multiple genes contribute to phenotype, leading to a range of skin color variation. Now the next question which is there, it is accumulation of phenylalanine in the body of phenylketonuria affected individuals. Why do the phenylalanine get accumulated? What happens usually is that phenylalanine which is there, my dear children, it gets transformed to tyrosine. You know, phenylalanine is an amino acid and it will transform into tyrosine when there is an enzyme, that is phenylalanine hydroxylase. Phenylalanine hydroxylase. When this enzyme is there, that's when this transformation happens. Now suppose the gene which was coding for this enzyme is mutated. Will this enzyme produce? No. And that's the exact thing that happens in phenylketonuria. In phenylketonuria, the gene that codes for this particular enzyme is not being, it's being mutated, which is why this enzyme is not produced. And if it is not produced, this guy won't transform into tyrosine. What will happen to this guy? This will be accumulated in the form of phenylpyruvic acid. Phenylpyruvic acid. Now this is a problem for us because it can cause mental retardation. And also one more thing, the tyrosine, if it is not produced, no, tyrosine is a basic requirement for formation of melanin and things like the pigments which is responsible for skin color and hair color. So if tyrosine is not being produced, side by side they will also have one more symptom, that is what? Their pigmentation. There is reduced pigmentation, okay, skin color and hair color, it will be hypopigmented because this is happening. So again over here this is an example of pleiotropic inheritance, okay. What do you call this as? Pleiotropy, where one particular gene, one gene is coding for two characters. One gene, basically the one that produces phenylalanine hydroxylase, it will produce two characters. One is, it will lead to accumulation of phenylalanine. Second thing, it is going to cause what? Reduced pigmentation as well. So one gene, two effects, that is pleiotropic characters. Okay, so this is the answer for this particular question. How do you write it? Let's see. Phenylalanine accumulates in the body due to defect in enzyme phenylalanine hydroxylase, which converts phenylalanine to tyrosine, causing toxic buildup in the form of phenylpyruvic acid. They basically get stored in the form of phenylpyruvic acid. Got it? Clear with this question? Now, define aneuploidy, give two examples of aneuploidy, mention the karyotype of Klinefelter syndrome of an affected individual and write its symptoms. So, chalo. Focused. What do you mean by aneuploidy? So basically, when when the gametes are being formed, the chromosomes, it divides. Okay, from a pair of chromosome, only one pair of chromosome will be only one guy will be going to each gamete. So if you're not able to visualize, let me help you with that. Okay, suppose this is A allele, this is B, chota allele. Okay, gamete formation is happening. One allele will go over here, another chromosome will be going over here. That's why we say that your gametes have half the number of chromosomes. So this half comes from the father and the other half will be coming from the mother. Now it will be forming diploid thing. But in the case of aneuploidy, there are chances that both of these guys came together over here. So now you have, instead of having one chromosome, you have two chromosomes. So there is gain of extra chromosome. And now one more thing, obviously when both of them are going over here, there is no one to go here. So there is loss of chromosome happening over here. Okay, there is loss of chromosome happening over here. This both gain and loss of chromosome, that is known as aneuploidy. Aneuploidy basically means when the chromosome number becomes different. How do you define it properly? Let me show you. So aneuploidy is a condition in which an organism has an abnormal number of chromosomes, either due to gain or loss of one or more chromosome. It is caused by non-disjunction during cell division. Non-disjunction is properly segregation is not happening, which is why this happens. So because of non-disjunction during cell division, my dear children, there is a condition that arises where there is abnormal number of chromosome. Either there will be gain or loss. So there are examples also. They are asking about example. One is Down syndrome, other is Turner syndrome. So in Down syndrome, what is happening is that basically from one parent you are getting one chromosome. Agreed. She had a pair. Suppose she had a pair. From this pair she got one chromosome. Now from another parent, both the chromosomes are coming over here instead of segregating. So now how many chromosomes do you have for one particular thing? 3. You have 3. So this happens in Down syndrome. in Down syndrome, the chromosome number 21, which should be coming like this from one parent, another parent. Here, there is one more chromosome coming from another parent, which is why I will call this as a trisomy condition. When one extra is there, I will call this trisomy of what? Chromosome number 21. Of chromosome number 21. Okay, this happens in autosomes. 21st chromosome is autosome, right? 23rd one is sex chromosome. So basically the sex of the child, if you are asked about the sex of the child, if from the mother it is X chromosome, if from the father it is Y chromosome, then this is a male child who is having Down syndrome. Sex chromosome will be different, right? Now similarly, if from the mother it is X, from the father also it is X, then it will be Down syndrome female. But anyway, this person will be having Down syndrome for sure. So trisomy of chromosome number 21 happens in the case of Down syndrome. Now Turner syndrome, if you are talking about Turner syndrome, what happens there? I told you, from one parent you get one chromosome, from other parent you are not getting anything. You are not getting anything, there is loss of chromosome. So this is sex chromosome, okay, this is X chromosome. Suppose from mother or father you got X chromosome, from the other parent you are not getting any chromosome right over here. So there is loss of chromosome, this is monosomy, there is only one chromosome. So I will say it is monosomy of 23rd chromosome. Or I will just call it monosomy, that's all. So both the cases are of aneuploidy, where there is what? Abnormal number of chromosome, my dear children. Now mention the karyotype of Klinefelter syndrome of an affected individual and write its symptoms. Now Klinefelter syndrome, what is that? Klinefelter syndrome as in, from one parent you are getting one chromosome, that is X chromosome, okay. From other parent you are getting X and also Y. Both the chromosomes are coming together. So how many chromosomes? You should be having just one each, right? But now the condition is where you have X chromosome, another X chromosome and another Y chromosome. This is XXY condition. This is what? XXY condition, my dear children. Okay. Now mention the karyotype. Karyotype as in number of chromosomes and everything. So usually we have 44 autosomes, that is 22 pair of autosomes, and then X and Y or X or X, either way. That is a usual number. That is a sex chromosomes. 45th and 46th. In total, we have 46 number of chromosomes. Okay. Now, how do you write it over here? Here, if you see, one extra X chromosome is there. So, I will write 44 autosomes. They are fine. But there is XXY condition. There is what? XXY condition. Or I can write like this. So 44, 45, 46, 47 chromosomes are there. So I can just write 47 XXY. 47 XXY. Either of these I can write for karyotype of Klinefelter syndrome. Now, what are the symptoms of this person, my dear children? So this had to be a male, so they would be having masculine characters, but they would also be having gynecomastia. That means there will be development of breast over there. Okay, and they will be also sterile, my dear children. They are not going to produce male gametes. They are going to be sterile. Their stature will be very, very tall. Okay, they are very tall. I'll show you. So over here, underdeveloped secondary sexual characters will be there. Gynecomastia, that is enlarged breast in the case of females. Infertility due to lack of spermatogenesis. Tall stature with long arms and legs, and learning difficulties in some cases. Now that is not mentioned in your NCERT, so I don't want you to remember this. At least this thing, if you can remember, that will be a lot more good. And this is the karyotype. You can write it like this, or you can write it like 44A XXY. Okay, either way. You can write this or this, that will be better. Fine. And with this, we complete that question as well. Where is it? Now, what is incomplete dominance? Explain it with reference to flower color in Snapdragon. So, how do you explain incomplete dominance, my dear children? Very easy thing. So, there is red color flower and there is white color flower. Now, this is the case of the flower's name is Snapdragon. Okay, what is it? Snapdragon. And the other name for this is, what do I call it? The Miraculous Jalapa. Just a second, okay? I think I have written, no? I think it's Mirabilis Jalapa only, but still I'll just confirm, okay? So anyways, this is red color and white color. It had two color flowers, okay? So over here we assume that red was the dominant one, because when we crossed this so pure red had capital R, and the white ones had small r. Okay, now this was the parent. Now gametes, if you form, this is the gametes, my dear children, always make it in round. So here also round. Okay, now suppose this and this fuses, you get capital R, small r. When you fuse this with this, you again get capital R, smaller. When you fuse this with this, you get capital R, smaller. When you fuse this with this, you get capital R, smaller. All of the cases, capital R will be dominant over the small r. So then all of them should be red in color. But in F1 generation, we found something odd. All of them were pink in color. They were not red, but they were pink. Why, ma'am? Because probably this was not completely dominant. This was incompletely dominant. Okay, it was not completely dominant over the small r. It was incompletely dominant, which is why there is a mixture of color, that is what? Pink. Okay, now obviously no one is going to be happy because this is not what we studied. This law of dominance where one is completely dominant over the other one. So we thought of doing an experiment. So crossed the two progenies up over here. So you have capital R, small r, and again capital R, small r. Okay. So over here the gametes are capital R, smaller, capital R, smaller. When you fuse this with this, you get capital R. When you fuse this with this, you get capital R, small r. When you fuse this with this, you get capital R, smaller. When you fuse this with this, you get small r, smaller. Now what was found? They found that one was red, two of them were pink, and one was white in color. That means if red was reappeared, that means if two alleles are coming together, that's when you get red. But if the alleles are like this, they're incomplete dominant, which is why it will still appear what? Pink in color. So what is the genotype of F2 generation, my dear children? Genotypic ratio. So two of them capital R, okay, one of them capital R, two of them capital R, smaller, and one of them smaller. So 1:2:1. What is the phenotypic ratio? Again, one is red, two of them are basically pink, and one is white. So this is also 1:2:2. Phenotypic and genotypic ratio, both of them are 1. Okay. So yes, it's not Mirabilis laipa, it is Antirrhinum something. Okay, that was the name of the plant. I'm so sorry. Mirabilis laipa, where did this come from as of now? Anyways, so red color flower, white color flower, you got pink flower, that is capital R, small r. And now in F2 generation, my dear children, again when you cross, you get all these things right over here. 1 red, 2 pink, and 1 white. So how do you write the answer? A phenomenon where both the alleles of a character express incompletely, producing an intermediate phenotype in the heterozygous condition, is known as incomplete dominance. Heterozygous condition as in both the alleles are different. Okay. Now Correns crossed homozygous red plant with homozygous white plant, and in F1 generation all the hybrids were pink colored. The dominant allele R was not completely dominant on the recessive allele small r, and this made it possible to distinguish capital R, small r as pink from capital R, red, and smaller white. When F1 plants were self-crossed, the F2 generation produced 25% of red flowered. Okay, that is 1:2:1, right? So 25% red flowered, 50% pink flowered, and 25% white flowered. From the hybridization result, we can observe that F4 phenotypic and genotypic ratio in incomplete dominance are 1:2:1. Okay, now just let me come for a second. I'll tell you the name. Yes, the name was Antirrhinum majus. That was the name, or majus, whatever you want to say, you can say. And the other name is snapdragon. Cool. So if at all the question comes like that, also you should be knowing. So they have not mentioned majus, that is the species name, but genus name is still mentioned in your NCERT, so you should be remembering it. Okay. Now write the schematic representation of one gene inheritance of Pisum sativum. What is Pisum sativa, my dear children? It is the guardian P, and you're talking about one gene inheritance, as in just one particular character in general. So basically I'll take capital T, small t, that is height, for gene for height. And how do I show it? I'll take pure plants. So this is parent, where I am going to take pure tall, and I am going to cross it with pure dwarf. So it is, did I do something? Yes, Jasmine did something. That is close it. So just a second. Keep, and then again open. It won't open like that. So what do? You do like this. Okay. No. Okay. A little bit of drama is very much necessary in my life, which is why it happens all of the times. Okay. These are the parents. Okay, how do you write it, my dear children? You write it as pure tall, as in they are homozygous. It will be capital T, capital T. Pure dwarf, as in it will be small t, small t. Agreed? Now the gametes. Gametes, you have to make like this, capital T. So this is a chromosome, a pair. From a pair, obviously one gamete will be having one chromosome. So either of them, whatever you do, it will be capital T only, because they are producing just one type of gametes. Similarly, it will be over like this over here. Now, if you're talking about F1 generation, what happens? So this crosses with this, you get capital T, small t. This crosses with this, you get again capital T, small t. This crosses with this, again capital T, small t. This crosses with this, capital T, small t. All of them capital T, small t, my dear children. That means all of them are tall. All are tall. Now what do? You try to find the F2 generation. How do you do that? So in F2 generation, this is F1 generation, right? In F2 generation, you have parents again, and this time it will be capital T, small t, and again capital T, small t. Heterozygous tall. You're going to cross it. This is known as selfing, because you're crossing between the progenies, right? Now gametes, if you see the gametes, how are they? Capital T, small t, and then capital T, small t. Cool. And F2 generation, what do you have? When this crossed with this, my dear children, you get capital T, capital T. When this crosses with this, you get capital T, small t. When this crosses with this, you get capital T, small t. When this crosses with this, you get small t, small t. Agreed? So if you see, ma'am, how many of them are tall? This is tall for sure. This is also tall because there is one capital T, two dominant traits. This is also going to be tall. So if you are asked about phenotypic ratio, if you are asked about phenotypic ratio, that means how do they look? The physical appearance. So 3 of them are tall, ma'am. And 1 of them will be what? Dwarf, ma'am. So 1 will be dwarf and 3 of them will be tall. If you are asked about the genotypic ratio, the genetic makeup, so 1 is capital T, capital T, 2 of them are capital T, small t, and 1 is small t, small t. So what is the genotypic ratio? 1 capital T, 2 capital T, small t, and 1 small t, small t. This is how you need to write your answers. Otherwise, how will your ma'am know or teacher know basically what is 1 represent, what does 2 represent, what is 1 represent again. So be specific with the answer, and this is a schematic representation of 1 gene inheritance in Pisum sativa. Done? Deal? Cool? Chalo. I hope you will be able to do it. Make sure you're drawing it properly, my dear children. Don't do hit, where the lines should be not be going down, okay, in one line. Both, for this allele, make the 2 lines here itself. This, and this. For this, you go that side. Okay, in that way you will be able to draw it properly. Chalo. Did we actually complete that chapter? Yes. Now we are going ahead with molecular base of inheritance, again of 13 marks, and it carries 13 marks, and it will be giving you two marks, 2 5-marker questions. So again, very important chapter, and little brainy chapter as well. So you should be knowing the concepts really well. Okay. So what we will do, we will drink some water and then we will continue, because even I am thirsty, and I am pretty sure that after listening to me for 1 hour 17 minutes, you might be not just thirsty but also hungry. So you can at least drink water as of now, or take a short break and come back really fast. 2-minute break is only fine. Okay, we cannot afford too much time. Okay. Your reminder to drink little amount of water, not little, at least too much amount of water. So before the class I drank lots of water, but now my now my voice has been cracking, which is why I drank it now. So let's start this particular chapter. Are you ready, my champions? So let's start with the salient features of double helix model of DNA. First thing, what is DNA made up of? DNA is made up of nucleotide. Okay, so now I am going to make something over here. You just have to see me draw that. Oops. Okay. This is known as a pentose sugar. That means it has 5 carbon. Okay, where are the 5 carbons? 1, 2, 3, 4, and the 5th one is like, I want to be outside. So this is the 5th carbon that is there. Okay. I'm making another pentose sugar right over here. Where shall I make it? I should make it here. 1, 2, 3, 4, and 5. Got it? Now listen to me. This is just a sugar. Now the sugar, when it binds with the nitrogen base right over here, my dear children, here also, where is it binding? It is binding on the first carbon. And what do you call this bond as? You will call this bond as glycosidic bond. Glycosidic bond. Agreed? Between the nitrogen base and the pentose sugar. Now the fifth carbon, my dear children, there is another bond that is happening. The phosphate groups are going to attach over here. There are phosphate, triphosphate thing. So what do you call this whole structure as? What do you call this whole structure? And remember, on the second carbon there is no OH group. So whenever there is no OH group on the pentose sugar, you will call this as deoxyribose sugar. You will call this as deoxyribose because it does not have OH group over here. Usually it has OH group. Okay, so this is deoxyribose and this is NB, that is nitrogen base. So together, if you see this and this structure, you call it as a nucleoside. Now, when there is phosphate group, you will call this deoxyribo triphosphates. Okay, there are 3 phosphate. So now one thing happens. First thing that you should know is this attachment that has taken place over here, that is phosphate group and over here the third carbon. Okay, you will call this as phosphoester bond. Phosphoester bond. Now what if I tell you that basically here there is some OH and from here to here there is some connection happening. So basically between the third carbon and between the fifth carbon there is some connection happening up over here. Then what do you call this bond? Again, this is between this and this. This is again an ester bond and again phosphate is involved. So I'll again call it phosphoester bond. But when this bonding and things are happening, my dear children, what happens? This bond breaks between the other phosphates. So initially it was deoxyribose triphosphate, but now when it is attaching, it has just become what? Deoxyribophosphate. That's all. So this is a nucleotide. This whole thing that you see is a nucleotide. This is another nucleotide that is being formed over here. Okay, and the phosphate bond which is there, it is broken, and this broken bond provides energy for the formation of other bonds, my dear children. Otherwise, the other bonds would not have been made over there. So deoxyribose triphosphates are the source of energy also, and they are acting as a substrate also. They are coming here, okay, they are providing energy as well. Remember that. Now since we have 2 phosphoester bonds, I'll just remove this name and I'll just put it, oh my god, I'll just remove the name and I'll just say it is phosphodiester bond. What is it, children? It is phosphodiester bond that is happening between the third carbon of a sugar and the fifth carbon of a sugar. Okay, so this is an glycosidic link and this is phosphodiester bond that is happening over here. Now the thing that you have to remember, this is a pentose sugar. What pentose sugar? Deoxyribose sugar. So usually there is an OH group. If there was an OH group, you will call this as RNA, but over here it is deoxy. Okay, so if there is OH group, you will just call it ribose. If there is no OH, you will call it deoxyribose, that is DNA, deoxyribonucleotides. Okay, nucleic acid basically. Now over here, listen to me. The nitrogen base over here will be having a complementary pair out there as well, and these nitrogen bases will be nitrogen bases will be forming bonds over here. It can be 3 bonds or it can be just 2 bonds. If it is 2 bonds, just understand here it will be adenine and there it will be thymine, or here it will be thymine and there it will be adenine, because adenine and thymine will be having 2 bonds, 2 hydrogen bonds. But guanine and cytosine, or cytosine and guanine, both of them will be having 3 bonds between them. So they are more tightly bonded. You can remember like that. And how do you remember it? G and C are kind of looking like each other, right? Because that's why they are tightly bonded to each other. You can remember like that. So there are hydrogen bonds between what? 2 nitrogen bases right over there. Cool, done. So now there is a strand like this, there is another strand like this. If you see, suppose this 5th carbon which is not attaching to anything up there, so you will call this end as 5' end. Over here the 3rd carbon is not attaching to anything, so I will call this as 3' end. So the DNA strand here, it is 5' to 3'. Similarly on the opposite end, it will be what? 3' to 5'. So they are anti-parallel to each other. They are what? Not just parallel, but they are oppositely parallel. So they are anti-parallel to each other. Now with the information that you have over here, we are going to write the salient features. Listen to me carefully. So over here, DNA is a double helix with two antiparallel polynucleotide chain. Polynucleotide as in nucleotides are coming together. So when the sugar and the nitrogen base comes together, you call this as nucleoside. When the phosphate also comes together, you call this as nucleotide. So polynucleotide as in nucleotide, all the nucleotides together, that is polynucleotide. Now the chains are coiled in a right-handed manner. They are coiled in a right-handed manner. So basically they are obviously coiled and they are coiled in a right-handed manner. You have to remember like that only. There's no other way. Now phosphodiester bond link nucleotides in 5' to 3' direction on one strand and 3' to 5' direction on the other strand. Okay, the bonds which are there, it is in 5' to 3', or basically the strands is in polarity. 5' to 3' polarity in one strand, other strand will be 3' to 5' polarity. Now hydrogen bonds between nitrogen bases hold the two strands. So adenine pairs with thymine with 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. Both of them, they are nitrogen bases, my dear children. Adenine and guanine, listen to me, adenine and guanine are known as purines. Okay, thymine and cytosine, they are known as pyrimidines. Listen to me, there is one more pyrimidine, that is uracil. But uracil is not found in DNA, it is found in RNA. In the case of RNA, you do not have thymine, but you have uracil. In the case of DNA, you do not have uracil, you just have thymine. Cool? Now follow Chargaff's rule. Purines is equals to purines. Whatever number of purines will be there, that much amount of pyrimidines will also be there. That means if adenine is 50 in one strand, and the opposite strand thymine also will be 50, because this is going to form a pair. Now if you are asked in one strand if adenine is 50, how much thymine is there? Array, in one strand, how do you know that? Adenine can be this much, thymine also can be that much. Thymine will be pairing with the other adenine over there. You are talking about one strand, things become a lot different. So do not forget that. Okay. Now one complete turn of a helix, that means suppose if it's like this. So this whole turn of an helix is, my dear children, I will call this is 34 Armstrong. Is what? 34 Armstrong. And this whole turn, my dear children, contains 10 bases. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So there are 10 base pairs in this whole turn. This whole turn, or this is known as pitch of DNA. This whole thing is known as what? Pitch of DNA, my dear children. That means one whole turn having 10 base pairs. And what is the distance? Basically, total thing is 34 Armstrongs. That is 3.4 nanometers. If you convert into nanometers, it will be 3.4 nanometer. Now, there are 10 base pairs. What if I tell you between each base pair, the distance is 0. Where is it? Or, since I am not writing in nanometer, I will just write 3.4 Armstrong. Okay, 3.4 Armstrong. That will be what? Distance between two adjacent nucleotides. So whole distance is 34 Armstrong. Between each of them, it is 3.4 Armstrong. So nanometer, if you are writing, you will write this as what? You will write this as 3.4 nanometer, and you will write this as 0.34 nanometer. Okay, 34, 3.4. Otherwise, 3.4, 0.34. We have to remember like that. Okay. Now, pentose sugar and phosphate form the backbone of the DNA. Obviously, sugar and the phosphates which are there, it is going to form the backbone of the DNA. Otherwise, it will be just stranded off. You need phosphate, you need two sugars that are going to form the backbone. So, these are the features that you should definitely write. Please make sure you remember nanometer and Armstrong and everything. Very, very important piece of information. Okay, cool, done, chalo. Now, DNA replication is fast, accurate, energetically expensive, substrate and enzyme dependent, initiated from a specific site and cannot uncoil on its entire length. Explain the process of DNA replication considering all these features. So, DNA replication is fast. How do you explain that? Basically, in the case of prokaryotes, the DNA polymerizes 2000 base pairs at second. So, basically 2000 base pairs per second is being replicated. If I use transcribed word, please forgive me, it's being replicated. That much amount of base pairs are being added up during replication in the case of prokaryotes, my dear children. So DNA replication works like this. Listen to me very carefully. Suppose this is a DNA strand. Okay. Now you cannot open the entire DNA. Why? Because there are bonds. because there are bonds, and to break these bonds you need high energy. You need what? High energy. So this is why it is energetically expensive. This is energetically expensive because of this reason, because there are so many bonds that it has to be broken down. So what do? You just open a segment of let's say DNA, and this segment will be known as replication fork. Replication fork. And now the DNA polymerase has to come and attach. Basically when you replicate, you break them first thing. The DNA is together, you have to break it. How do you break it? You break it with the help of DNA helicase. There is an enzyme that will be breaking down the DNA into two segments. Okay, so obviously it is enzyme dependent because it is taking enzymes help. So first enzyme is DNA helicase, it will be opening up, and now my dear children, there are certain proteins also which will be coming and binding over here. Why? Because you don't want them to coil back again. So these are single stranded binding proteins also which comes and binds. Now what happens? Slowly, there is a site over here in the DNA replication tract that is known as ORI site. That is known as what? ORI site. ORI site. So it is accurate. Okay, and it is initiated from a specific point. This is the point. It is initiated from the specific point, and that is known as ORI site. Now, the DNA polymerase, the next enzyme will be coming in and will be synthesizing the DNA in what direction? 5' to 3' direction. Suppose this strand is in 3' to 5', then from here itself, the next strand that will be formed here will be in 5' to 3' direction, my dear children. So obviously it is accurate in that sense, and it makes sure that it is not making any errors over here. It is not making any errors over here. Now for making a new DNA strand, okay, obviously what do you need? You need substrate, and what is that substrate? I told you DNTPs, that is deoxynucleotide triphosphates. Okay, you need triphosphate, deoxynucleotide triphosphates, which are 3 phosphates. Those are the substrates that you need, and now these substrates will be joined together to form what? A polynucleotide chain. Now how do you write the answer? This is just a basic thing that I explained, right? How do you write the answer, which includes all the points? So begins as a specific origin of replication. Now that is a point, initiated at a specific point. Now helicase unwinds the DNA only at specific site and are forming a replication fork, because uncoiling the whole DNA is energetically expensive. Now we've included the next point, energetically expensive point. Single strand binding proteins stabilize the unbound DNA. Primase synthesizes primers to provide a starting point for DNA polymerase. Yeah, that is also there. Okay, so when DNA is going to replicate, the primase is going to come and add a primer, because DNA replication cannot start off like that. So you need a primer over here. So again you need enzymes. How many enzymes? Helicase, primase, all of them. Basically that means this particular replication process is what? Enzyme dependent. This shows that it is enzyme dependent as well. Now elongation, DNA polymerase synthesizes the new strand in 5' to 3' direction. Okay, it is very accurate in that sense. Leading strand is synthesized continuously. That means the strand which is in 3' to 5' direction, here the opposite strand will be 5 to 3 and it will go smoothly. But what about the other strand which is in 5' to 3' direction? Here the formation cannot happen this side, right? The formation will be happening this side. But the DNA is uncoiling. So obviously the DNA polymerase cannot go that side, because it is going to come this side, my dear children. So how can it go that side? So again a primer have to be added there. Every time a strand is being opened, my dear children, over here, what is happening? Suppose this is the strand. It was like this initially. So this is 5', this is 3', this is 3', and this is 5'. Here it's fine. Here it is going on like this only, in this direction. But what about this side? Every time it will open up. Okay, suppose this have opened up a bit more. So already a primer was added over here, and DNA polymerase was extending this side. Now again you have to add a primer over there. Again DNA polymerase has to extend. If you see, this is a lagging strand where it is not continuously happening, where DNA is not polymerized continuously. This is a leading strand where DNA polymerization happen continuously. So leading strand is synthesized continuously, lagging strand is synthesized in short Okazaki fragments. Now these are fragments that are known as Okazaki fragments, which are joined together later with the help of DNA ligase. Now there is a point that is accurate. So proofreading by DNA polymerase ensures accuracy. You know what? Every time DNA adds something, it has to make sure it is a correct base pair that is being added up over there. So again DNA polymerase will check the whole thing, that is proofreading has been done by DNA polymerase. Now this shows what it is very much accurate. It is substrate dependent. How do you write it? You can write it in this point itself. You need deoxynucleotide triphosphates. Deoxynucleotide triphosphates for making a chain of nucleotides. Otherwise, how do you make a DNA? So that is there. Now termination occurs when replication forks meet. So when suppose from here also replication is happening, here also replication is happening. So when two replication forks meet, that when termination can happen, or it reaches the termination sequences, that's also when it happens. Now enzyme removes the RNA primers. So everywhere wherever the RNA primers, you will remove it because this is a DNA strand. And then another enzyme, that is DNA ligase, will be ligating this whole thing. So DNA ligase seals the final nicks in the backbone. Final gaps in the backbone has been treated by the DNA ligase. So having included all the point, it is fast. Yes, we already told about it. It's accurate. It is energetically expensive because you have to break down lots of bonds. Substrate and enzyme dependent, you need lots of enzymes. Initiated from a specific site, yes. Cannot uncoil on its entire length, yes. Cannot uncoil on its entire length. Obviously, you need enzymes for that. Get it? Done. And you have single stranded proteins as well, which is why they would not be coiling back. Now explain the process, we did it. So these are the things that you have to definitely mention. That's how you get marks. Now given below are sequences of nucleotides in a particular mRNA and amino acids coded by it. So this is the mRNA that you have in the 5' to 3' direction, my dear children. So AUG, UUU, UUC, so lots of things are being given. Write the properties of genetic code that can be correlated from the above given data. So when this type of question comes, you have to obviously look for the properties. Apart from that, whatever you remember about the genetic code, you have to write that as well. So the first thing that has been very much visible over here, that AUG is coding for methionine. Now AUG, if you remember, it's a start codon and it is also coding for methionine. That means it is showing dual function. So dual function by AUG, first thing. Now UUC, there are two codons coding for the same amino acid, that is phenylalanine. What do you call this feature as? This feature is degenerate feature of amino acids. Sorry, codons. So codons are degenerate because two different codons or multiple codons codes for what? One amino acid. Multiple codons codes for one amino acid. Now apart from that, if you see GAG is there, UUU is there, or UAU is there, GUG is there, do you think that there is any punctuation? No. So it is what? It is continuous. It is continuous. There is no punctuation. No punctuation is there. And if you see, you do not see any repetition. Okay, you do not see any repetition. One particular codon is going to code for one particular amino acid, isn't it? So this is unambiguous nature of the codons. One codon for one particular amino acid. Okay, one codon will be coding for that particular amino acid itself, not for any other amino acid. So one codon codes for one amino acid. Now if you see UAA, it is not going to code for any amino acid. That means UAA is what? A stop codon. So there are stop codons as well which does not code for any amino acid. So these are 1, 2, 3, 4, 5 features that I only found out. Now you can think for your own things also, but let me tell. Okay, so the codon is triplet. Yes, if you see all the codons, my dear children, they are in triplet format. Yes, 3, 3, 3. 61 codons code for amino acids and 3 codons not code for anything. They are known as stop codons. Okay, so total 64 you have. Out of them, 61 are going to code for amino acids and 3 of them are not going to code for anything. Now AUG has dual function. It codes for methionine, okay, and it also acts as initiator codon. Now some amino acids are coded by more than one codon, hence the code is what? Degenerate. Now the codon is read in mRNA in a contiguous fashion. There are no punctuations in there. Now UAG, UGA are stop codons, terminator codons. Okay, they are UA, UGA and UAG. They are terminator codons. Now the code is nearly universal. For example, from bacteria to human, UUU would be coding for phenylalanine. That is an extra feature. That is not being mentioned over there, but yes. So on all organisms, the codons will be coding for the same amino acid. Now some exceptions obviously will be there in mitochondrial codons as well as in some other organisms, that is protozoans and stuff like that. Works? Cool? Chalo. So this is right. This is what you write about codons. Now, just ma'am is tired, so I have to energize myself. Just like how you need motivation, I also need motivation. So I'll say, Jasmine, you can do it. Yes? So children, you can also do it. Let's do it together. So list down or describe the steps involved in DNA fingerprinting technique. What do you mean by DNA fingerprinting? Just like how we have fingerprints, which is very unique to us. Similarly, in our DNA, there are certain segments which are very unique to us. And you call them variable number of tandem repeats. Okay? So these repeats, there are certain repetitions which you have in your DNA. For example, a codon A, GCCCA, GCCA. So this might be repeated 50,000 times in my body, but this is very unique to me. This is not your sequence. Your sequence will be different. Okay, so I am going to utilize this particular feature, and I am going to take into consideration to find criminals and things like that. How? Let's see. So I have explained it so many times, but still, since it's a 5-mark question, we have to explain it again. So my dear children, suppose there's a crime scene that has happened. Over there is blood sample. Okay, there is some blood sample. Blood sample. Not of the person who was killed, but probably the suspect. Okay. Now I am the suspect, and there is my friend Chintu, who is also a suspect. Okay. So basically what is done is that blood sample is collected. Okay. You take out the DNA, and now what they did was, they made a complementary strand of this particular DNA. They know the DNA strand, they made a complementary strand of it, and they have tagged it with something, which is why it is known as a probe. So it will show up in UV condition and things like that, or under autoradiography, whatever. So it's a probe. Okay. Now I and Chintu, both of our blood samples were collected. Okay. What was done was, again, my DNA was taken, isolation of DNA was isolation of DNA was done. That is, it is separated from proteins and everything else. And then the DNA has been cut with the help of restriction enzymes, my dear children, with the help of particular enzymes, which will be cutting at only those particular sites. So where G and C and everything will be there. So similarly, mine and Chintu both were cut. Okay. So there was gel electrophoresis that was being done after that. The DNA was run. So you saw a strand like this. Oh, I should not be making in orange. Wait. So first is isolation, then you cut the DNA, and now you're running through, what say, this thing. Gel electrophoresis. Now this is mine and this is Chintu's. Okay. Now what happened? Basically, it's on gel electrophoresis. Now you take a nitrocellulose sheet, you transfer all the DNA on the nitrocellulose sheet. Now I have a sheet like this. Suppose this is a sheet now. The next thing that was done is, the probe which was there, it was made to run through this. What will happen? I have the sequence for this, Chintu does not, because it is my DNA strand. So I have the complementary strand of it. So what will happen? This particular probe will be going and attaching on mine. Now under the autoradiography machine, there will be visibility that, hey, this is the DNA where it is going and attached. Whose is this DNA? And that's when they come to know that it's mine and I will be jailed. So this is the use of DNA fingerprinting technique, my dear children. I hope you got it. Now how do you frame the answer for this? You frame it like this. Isolation of DNA. DNA is extracted from the sample, that is blood, hair, saliva, whatever is there. Now digestion by restriction enzymes. DNA is cut into fragments using specific restriction enzymes. So sites will be cut. Okay. Now separation separation by gel electrophoresis. DNA fragments are separated based on size using the agarose gel electrophoresis. Now transfer to membrane, that is nitrocellulose membrane. This is known as southern blotting. The separated fragments are transferred onto the nylon or nitrocellulose membrane. Now hybridization with DNA probes. Radioactive or fluorescent probes which are there, complementary to the specific sequences, bind to variable regions of the DNA. Obviously, yeah. Visualization. The hybridized fragments are visualized using X-ray film or UV light, producing a unique DNA pattern. This pattern is compared to the identity or relationship. Now you can do the same thing for not just criminal things, okay. You can find for paternity issues, for maternity issues also. If you have the probe, that means basically the child, it's yours. You get it? Got it? Done. So this is DNA fingerprinting, my dear children. And always remember, from parents to children, obviously there will be some similarity. It won't be like two different people. Okay? So that is why we are able to find the parent and children relationship as well. Going ahead. Answer the following. This is a very fun question, always asked. So, construct a complete transcription unit with promoter and terminator on the basis of hypothetical template strand in the given below. Write the RNA strand transcribed from the above transcription unit along with polarity. Now, my dear children, you are asked to make a transcription unit. Okay, you know, in transcription unit, what is there? There is your DNA. Okay. There is a DNA, and over here template strand is given. That means the strand which is going to undergo the process of transcription is given. That is known as template strand. Template. But just to confuse you, they had did something. So this is 3' end. Wherever the arrow goes, so over here the arrow is going towards this side. That means it is a 3' end, and this will be the 5' end. So ma'am, is it okay if we put it like that? Yeah, it's okay. Why? Transcription will be happening in 5' to 3' end. Transcription, the new strand will be formed like from here to there. So it's fine, totally fine. Okay. So, so now over here, I am putting all the sequences A, T, G, C, A, T, G, C, A, T, A, C. Okay. Here, this will be the coding strand. I am talking about DNA, my dear children. This is a coding strand. This is a pair of, let's say, the total helix. Okay, one strand, complementary strand. So complementary strand will be having complementary codon as well. A will be going with T, T with A, G with C, C with G, A with T, T with A, G with C, C with G, A with T, T with A with T, and C with G. Okay, what is the polarity? This is 5' to 3'. Agreed? Now, still it is not a complete transcription unit. Why? Because RNA polymerase which will go do the transcription, no, it needs a promoter site. So there will be a promoter site over here towards this side, that is towards the 5' end of the coding strand. You remember like that. 5' end of the coding strand, that's when there is a promoter region. There is a promoter region onto which the RNA polymerase will come and bind, and that's when the whole process will happen. Similarly, over here also, there will be a terminator region. When it reaches over here, transcription will be stopping. Now, this is what a transcription unit, my dear children. So, construct a complete transcription unit with promoter and terminator on the basis of hypothetical template strand given below. This is what you get. Now, write the RNA strand transcribed from the above transcription unit. Now, you have to do the transcription and tell what strand is going to be made. So, I will tell you an easy thing. So, wherever there is T, you can replace it with U. Because anyway it will be complementary. The RNA which is formed, that will also be complementary, but RNA does not have thymine, it has uracil. So the strand that will be made, my dear children, will be something like this. Don't make it inside, okay? I am just making because for easy work. So here there is T, it goes A, right? No. So wherever there is T, my dear children, it will be forming U. It will be forming U. So A will be going with U instead of T. Here it will be A itself, here it will be C itself, here it will be G itself, here again it is T, so that means it will be U. Here T is there, so again it will be A, okay, C, G, again here A is there, that means it will be bearing with U, not T. Again A is there, it will be, so here A is there, A will be going with again U, not T, and here it will be G. So this little bit congestion there, that's why it happened, but yes, this is the strand you get. So if you see, wherever you had T, you are replacing it by U, rest everything is same. And the polarity is very much important. So this will be from 5' to 3'. This is the mRNA that you get. Not make it inside, I am telling you again, you have to make something like this. So this is the one that I typed, this is more easy. Okay, will you be able to remember this? Yes, ma'am. So what's the problem when you just copy this down and you replace it with U? The problem is if you make a mess around over here, if you make a wrong statement, wrong codons over here, wrong complementary strand over here, you are going to mess it around here as well. So please double check and then only make it. So this is the transcription unit, this is the mRNA that you get after transcription. Works? Cool? Chalo. Next question. What are the salient features of human genome? So how do you write the salient features of human genome? Let's see. These are the salient features. Human genome contains 3,106.7 million base pairs. Okay, one thing. Now, the average gene consists of 3000 base. If you're talking about gene, my dear children, a gene will be containing what? 3000 base pairs, average ones, okay? But sizes obviously vary, with largest known human being gene as dystrophin. That is a gene, my dear children. That means a protein. A protein is already coded by a gene, so you will say that this is a dystrophin gene. Okay, now it has 2.4 million bases. So average is 3000 bases, but this has 2.4 million bases. Now total number of genes is estimated to 30,000, much lower than previous estimates of 80,000 to 1,40,000. Only that much amount of genes we have. The human genome, as in the whole genome, 3,164.7 million base pairs are there. Now average gene size is 3000 base pairs, but over here in dystrophin, it is 2.4 million bases, okay, 2.4 million bases. Now, total number of genes which are there, it is 30,000, but it was thought it was 80,000 to 1,40,000, okay. Now, the functions are known for over 50% of the discovered genes. So, we know 30,000 genes. Out of that, only 50% of genes function has been actually known. Now, less than 2% of the genome codes for protein. If you talk about the whole genome, only less than 2% of the genome actually codes for some amount of protein. Rest will be what's just unwanted sequences, like repetitive sequences. They are repetitive sequences which make more large portion of the human genome. Repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1000 times as well. They have no direct coding function, but they shed light on chromosome structure, dynamics and evolution. Now chromosome number 1 has most genes, that is 2968 genes. Okay, that many genes are there. And Y has fewest, that is 231. So 2968 and 231, this is chromosome number 1 which is highest, and Y which has the fewest, that is 231. Now scientists have identified about 1.4 million locations where single base DNA differences are there. That means only at single base pairs we are different. Rest everything is same. That is 1.4 million locations, okay. That is known as single nucleotide polymorphism, my dear children. So these are the features. Obviously there is no much thing to understand and all. You have to remember it. So there is no other way. Write it down somewhere and try to remember it, okay. That's all. Next question, describe lac operon concept of gene regulation. I would be explaining it, but now I think there's high time for some break. It's already almost up to 2 hours. Okay, my voice is cracking, I'm tired. So I'll just quickly take a break and come. I request you all to take some break and come back again. Okay, that's all. Chalo, we'll see. Hello champions, I hope you're back from your break. So let's start. We were talking about lac operon. So we have to describe the operon model of gene regulation. How do you explain it? Basically, just understand this particular logic. So for every gene that you see, my dear children, you have a promoter region onto which what will come and bind? RNA polymerase will come and bind and do the whole transcription. Now suppose, okay, in this particular gene, there is one more region which is known as operator region. And what is the use of this operator region? Basically, what happens is that on this operator region, if for suppose, okay, suppose one inhibitor is coming and attaching over here, a repressor protein is coming and attaching over here. Now repressor protein, as in it is going to repress something, it is going to do some negative activity. So when a repressor protein comes and attaches on the operator region, what happens? The RNA polymerase which came and attached over here would not be able to move forward, and it won't be able to transcribe the genes which are over here. So in the case of lac operon, the thing is the same. Basically, they have a structural gene which looks like this. I'm going to zoom it and show. So ZYA, it is a structural gene. Z codes for beta-galactosidase, my dear children. Y codes for permease and A codes for trans-acetylase. What is the use of beta-galactosidase? The use of beta-galactosidase is to break down lactose. Lactose is a component, okay? Lactose is a form of sugar which basically when you break it down, you get energy. The organism can take out energy with the help of it. So when the lactose is there, it breaks them. Beta-galactosidase which is there, it is going to break down this lactose into two. That is how the whole organism is going to get the energy. Now permease, what is the role of permease? Suppose this is an E. coli. E. coli is a prokaryote, right? Now you want the lactose to enter the E. coli. For that you have to make the membrane permeable. For that you have permease, my dear children. Now trans-acetylase has some other functions, which is, for example, dealing with the byproducts which are going to be formed in this particular process and things like that. So this is a structural gene. Now before this you have an operator region, before this you have a promoter region. Now this is about one whole gene. Now over here if you see there is one more gene and it has one more promoter sequence as well. But this gene is known as regulatory gene. It is the one that is going to regulate the process. So it can either code for a repressor protein my dear children, or basically it can code for an activator protein. But over here we are talking about a repressor protein. Suppose this repressor protein is there, it goes and binds on the operator. Now even if RNA polymerase want to transcribe it, it won't be able to do that. So I'll say the lac operon which is there, it is in a switched off condition. As of now, it is totally switched off because there is no transcription that is happening over here. But let me tell you, little amount of transcription happens always. Little amount of transcription happens always, but not in a full swing. Now, if you want transcription to happen, what would you do? Basically, in the presence of lactose, when lactose itself is there, my dear children, what will happen? The lactose will come and bind on this repressor protein. It will bind on this repressor protein. As you can see over here, inducer is nothing but lactose itself. Lactose or allolactose, when it comes and binds on the repressor protein, my dear children, this repressor protein would not be free to come and bind over here. Hence, if it is not free, the transcription can easily occur and the lactose which is there, it can be broken down easily. Now, your question, ma'am, if permease everything was not being formed, then how did lactose actually enter? So I told you, little amount of transcription always takes place. So with this little amount of transcription, permease lets the lactose inside when lactose is there. So lactose is going to act like an inducer, my dear children. So it is negative regulation. Why? Mostly it is negative regulation because repressor protein binds over there and represses the whole process. You get it? Now, how do you frame this answer? Ayo, yes. So, switch off mechanism and switch on mechanism is there. In switch off, when lactose molecules are absent in the nutrient medium, DNA is switched off for protein synthesis. There is no transcription happening over there. Okay, that's what we say, but a little amount of transcription is still there. Now, the regulator gene produces a kind of protein called repressor that binds to the operator gene. This do not allows RNA polymerase enzyme to transcribe the structural genes. Hence, there is no transcription, translation, and the required protein is not being synthesized. Hence, this is known as switched off mechanism. Now, on the other hand, switch on mechanism, the lactose molecules added in the nutrient medium acts as inducers which bind to the repressor to form inducer-repressor complex. Now, the repressor becomes inactive and released from the operator region. This enables the RNA polymerase enzyme to transcribe what? All three structural genes, that is Z, Y, A, ZIA. Okay. Now, all three genes mRNA strands undergo translation to produce 3 polypeptide chains or 3 proteins which acts as 3 enzymes, that is beta-galactosidase, permease and transacetylase. These 3 enzymes together break down the lactose into what? Glucose and galactose. And the whole story ends up right over here. So, you should be knowing about this funda. If you do not know this particular thing, you might not be able to answer. Cool. I hope it's clear. Paka clear. Let's get into the next topic. That is, DNA replication is said to be semi-conservative. Why? Describe the experimental proof of Michelson and Stahl to show DNA replication is semi-conservative. So, what did they exactly do? Basically, they had their DNA. Okay, you know DNA has nitrogen bases and things like that. So basically nitrogen is a component of DNA. Now there are different isotopes of nitrogen, heavy isotope, lighter isotope and things like that. So what happened? Basically for once E. coli was being grown in a medium which was rich in the heavy isotope, that is N15 isotope. Okay, now it was grown in this particular medium. Obviously this E. coli will be having the DNA which will be made up of what? This N15 nitrogen isotope, right? So after that, if you centrifuge the whole thing, you will find that there is a heavy band that is being formed over here, and since it is heavy, it is moving there and getting accumulated over here. If you put the whole thing in cesium chloride, okay, if you put the whole thing in cesium chloride and you allow it to sediment, what will happen? The whole thing will be sedimenting towards down. So there is a heavy band that is being formed. And why is it heavy? Because both the strand of DNA, my dear children, will be having N15 nitrogen base pair, isotope. Okay. Now what you are doing is that to understand semi-conservative nature, what do you do? You are going to change the medium into N14. You're going to change the medium into N14. So what is the major thing that you can observe over here? When you change into N14, what is the major thing that you observe over here? Now suppose the E. coli is going to divide into 2. Suppose the E. coli divides into 2. So over here, the DNA which is there, it also has to replicate. So it will open up. This strand opens up, my dear children, and now it is N14 medium. So what will happen? A new strand is being formed over here and a new strand is being formed over here. Both these new strands has what? N14. You know replication is semi-conservative. What do I mean by that? That means basically if this is a strand of DNA, when a new strand of DNA forms, both these strands would be there, parental strands will be there, and there will be a new strand that is added up over here. Similarly, these were the old strand, the parental strands. The parental side, they moved apart, okay. they moved apart, and now the new strand is being formed. New strand has N14, my dear children. But how do you prove it? How do you prove this particular thing? So over here, one E. coli has this, okay, and the other E. coli again will be having this, and now you're going to put it in the medium, and you're going to see that basically there is one band which is being formed, and it is a hybrid band. It is somewhere in the middle. It is not towards deeper side. It is somewhere in the middle. That means basically what happened, there is both N15, there is both N15 as well as N14 present over here, which is why it is creating a hybrid situation right over here. Now this is when you did it for 20 minutes. After 20 minutes, E. coli will be keeping on dividing. So the next 20 minutes, my dear children, that means after end of 40 minutes, what will you find? Again, this strand which is there, my dear children, it is going to open up. There will be a parental strand. These two are the parental strands now. Again, you are putting in N14 medium only. So what will happen? There is a new strand being formed. There is a new strand being formed. The first time you see both the strands are N14, N14. But this will be what? N14 as well as N15. So you are going to see hybrid strand as well as what? A lighter strand, because it is made up of both N14. Similarly over here also you would be seeing the same thing. So there will be one which are totally N14, N14. There will be two of them where N14 and N15 will be there. Just like that mentioned over here. You get it? So this time what will happen? You will find an hybrid and also you will find light ones. So why are you able to find? Because the parental strand is still there and you are semi-conservative. You are conserving it. That is why it is known as semi-conservative method of replication. You are conserving it. One strand is being conserved. Another strand is what? Totally new. Which is why you are getting a data like this. This is the Michelson-Stahl experiment, my dear children. So 20 minutes, after 20 minutes, that is after 40 minutes, you get this. Now again, if you keep it for 20 minutes, again you would be seeing that these strands would be more in number, N14 would be more in number, but there will be two strands definitely which will be having one N15 strand. Okay, two strands will be there which will be just like this, because again when it divides, it will be forming like that only, right? Here also. So I hope you got this. So this is Michelson-Stahl experiment that you have. So you just go through all this thing. It is really important for you. You just have to go through, okay? And do not explain in this big format. You do not have time for it. So look for the crisp and short content. Just make shorter content out of it and write it down on the day of your exam. Now next thing, Oswald, Avery and others have continued Griffith's transforming principle to prove DNA as genetic material, substantiate. So basically, my dear children, Oswald, Avery and everyone, they did, so Griffith was a person who did an experiment, okay. According to his experiment, what was happening, there was a strain that is dead S strain, my dear children. There was dead S strain and there was live R strain. Okay, heat killed S strain and live R strain, and basically when he put it inside the organism, okay, inside the organism, that is basically mice, he found that this mice was still dying. This mice was still dying, my dear children. Why was it happening? Because probably when both of them were mixed over here, the R strain which is there, it is getting converted to something else, because something from here it is getting transferred to the live R strain and making it what? S strain. And you know what? S strain is a virulent form of strain which is going to kill the mice. So what was this transformation? That was the whole question about. And Oswald, Avery, MacLeod, McCarthy, everyone were working on it. They wanted to, Oswald, Avery, that was one name, okay. Oswald, Avery, MacLeod and McCarthy, all of them, they were working on it and they wanted to know what is that something that is getting transferred. So what if I treat both of them, okay, what if I treat this mixture with protease? If I treat with protease, what will happen? Basically, it will kill down proteins. It will kill down proteins. Now, if it is proteins are being killed down, still is the transformation taking place or not? Then they found out that yes, transformation is still taking place. Then they tried RNAs. They broke down RNA, my dear children, with the help of RNAs. Still they saw that yes, transformation is still taking place. Then they treated with DNAs. When they treated with DNAs, my dear children, when the DNA was getting damaged, that's when the transformation was not taking place. And that's how they got to know that DNA is obviously the genetic material, because when this is damaged, that is when transformation is not taking place. When it is there, that's when probably the DNA is moving from here to here and making this live R strain turn into what? Live S strain, and making it, making the mice infected, and later the mice dies. So this is the whole thing about. But still, people didn't believe it until other people came in, that is Hershey and Chase came for their experiment. Until then it was thought that something is there which is acting as genetic material. We do not know what is it. So proteins, RNA and DNA was there. They gave a clue that it was DNA, but still people didn't agree. For that they had to do another experiment, that is Hershey and Chase experiment. There were 2 other people, but yes. So this is what you have to write. Heat killed S strain which was there, my dear children, and plus protease plus R strain, transformation was still taking place. Heat killed S strain plus RNase plus R strain, still transformation is taking place. Heat killed S strain plus DNase plus live R strain, no transformation was taking place. So protein of heat killed S strain is not genetic material, as there is transformation of R strain into S strain in the absence of protein. RNA of heat killed S strain is not the genetic material, as there is still transformation, right? DNA of heat killed is the genetic material, because DNA digested with DNase mixed with R strain is unable to transform the R strain to S strain. So you might feel that, ma'am, what are you saying? But when you read it, or basically if you understood the whole concept, then it becomes a lot more easy for you to understand it. Okay? Okay. Fine. Going ahead, explain the process of translation in protein synthesis. So what exactly happens in translation? You know, there is mRNA. We know one thing. There is mRNA. And on this mRNA, there are few codons like AUG, GUG, some things like that. And at the end, it is UAA, some stop codon. Now, this is in 5' to 3' direction. Okay. Now, these codons which are there, my dear children, exactly complementary anticodon will be there on the tRNA. So you know tRNA, in the tRNA this is anticodon loop which will be having the anticodon, and this is the 5' end, this is the 3' end. On the 3' end there will be an amino acid that is being attached. So basically the first thing that happens is, in the first phase of translation, the amino acid will come and bind on the tRNA. This is known as amino acylation. What do you call it? Amino acylation, or you also call it charging of tRNA. You call it charging of tRNA, my dear children. After that, what do you do? Basically, the charging is done. After that, the mRNA which is there, it will slowly bind with the small subunit of the ribosome. With the small subunit of the ribosome, this particular thing will come and bind. And that's not all. The next thing that comes is what? The tRNA. The tRNA, my dear children, I am going to make like this with the amino acid and also the larger subunit right over here. So there are certain sites on the larger subunit where the tRNA will be coming and binding and things like that. Okay. Now over here you have AUG, GUG, UAA, everything. Okay. So AUG is the codon. Anticodon will be what? Anticodon will be what? U, A and C. You get it? So this is how, again slowly, again one more tRNA will be coming in. Again one more tRNA will be coming in, forming a bond over here. Then these amino acids which are there, they will form peptide linkage with each other. They will form peptide bond with each other. What happens when it forms peptide bond? Basically, they bond the break over here. So now the amino acid is free to be attached to here. Similarly, another amino acid comes and this goes on. The chain formation goes on until the terminator sequence is being reached, where it is not going to code for no tRNA is going to come over here, because here the termination has to take place. Stop codon does not code for any amino acid, so no tRNA is going to come and attach over there. Got it? So how do you frame the answer? So translation is the process of synthesizing a polypeptide from an mRNA template. This process of translation takes place in the cytoplasm of a cell, okay, where ribosome is located. So obviously remember, translation is going to happen in the cytoplasm because you need ribosome. Ribosomes are the one which is going to help in attaching and reading and everything. So activation of charging of tRNA or amino acid. So amino acylation of tRNA is the process of attaching an amino acid to its corresponding tRNA. This is also known as charging of tRNA. This happens. Then initiation happens. mRNA binds to the small ribosomal unit. The start codon is recognized by initiated tRNA carrying methionine. The large ribosomal unit assembles, forming the initiation complex. So basically first this, then as I said, the tRNA with the codon, the anticodon for this, that is the methionine. This is start codon as well. Okay. Then the larger one also assemble, and now you have the complete complex, right? So elongation. tRNA brings amino acid to the ribosome, matching codons on mRNA through its anticodon. Okay. Peptide bonds form between the adjacent amino acid catalyzed by peptidyl transferase. So it is catalyzed by what? Peptidyl transferase. Now ribosome moves along the mRNA in 5' to 3' direction. Ribosomes would be moving from 5' to 3' direction, my dear children, in this direction. It will go on. The ribosome itself will be moving. That's how the different sites and everything it is open. Okay. Now, termination, when a stop codon is reached, a translation stop, as the release factors binds to the stop codon, freeing the newly synthesized polypeptide. So, there is no tRNA that comes and attached, but the release factors will be coming and attaching and releasing the tRNA and the whole strand over there. Got it? And that's how you get a new protein. Fine. So, this is it. Now, describe Hershey and Chase experiment. I think this is the last question from this particular chapter, isn't it? Yes. So, over here, my dear children, what is Hershey and Chase experiment? I told you to confirm that DNA, yes, DNA is the genetic material. Hershey and Chase experiment was being done. So, over here, two things that you have to notice. The thing that you used to infect the E. coli was bacteriophage, and bacteriophage is a virus. Bacteriophage is a virus that is there to infect the bacteria, right over here. Now, viruses caspid, no, the outer layer, it is made up of protein. It is made up of protein. So, what did you do? Basically, you gave radioactive S35 labeled protein capsule. So, there was S35 being included over here, and this S35 is radioactive labeled thing, okay. Now, you are going to put it over here. So, it's there, it is incorporated into the protein, So, wherever there is protein, you will be finding S35 there. Now, talking about this particular bacteriophage, here you are going to, you basically put radioactive P32. Radioactive P32 labeled DNA. So, phosphate is obviously a very important structure of DNA. So, this particular phosphate was even taken up, and hence the DNA is the one that is radioactive over here. Here the protein is the one that is radioactive, that is the outermost layer, and here the DNA is the one that is radioactive. Now, what did you do? You basically try to infect it. When you infected it, the bacteriophage will be transferring the genetic material, my dear children, and rest of the things would be left off. Rest of the things which are there, the capsid which is there, it is left off. Okay, the genetic material is being passed. So, they did some sedimentation. Okay, they did they blended it properly, and then they centrifuged it. They shook it in high speed. Okay, and then they found out that there was no radioactivity detected in the cells. So, the cells which was there, they did not find radioactivity in the cells, but the supernatant, okay, the supernatant which was there being collected, my dear children, in that they found radioactivity. That means the genetic material does not contain protein. If the genetic material contained the protein, then the cells which was there, it would have been radioactive. But the genetic material does not contain protein, because protein was the one that was being tagged, and protein was not visible in the cells. On the other hand, basically, when this infection was done, the genetic material was passed over here inside the E. coli. Now, when the cells were separated, it was found out radioactivity was detected in the cells. That means it is the DNA that is being passed on. It is the DNA that has been passed on, and no radioactivity was found in the supernatant, my dear children. So, here you know that DNA was the one that had the radioactive ability, because we put phosphate over there. Okay, so if the cells are radioactive, that means it is the DNA that has been transferred. Here, since you got to know the cell is not radioactive, the supernatant is radioactive. That means what was there in the supernatant? It was the protein that was there in the supernatant. You get it? So, that was not it. So, this was the whole experiment of Hershey and Chase, and with this they proved that protein is not the genetic material, but DNA is the genetic material. Again, how do you need to write? So, labeling the phages, group phages in two separate medium, okay, one with radioactive S35, always remember 5 looks like S. So, 35, S35 is for radioactive S, okay. Now, 35 is for S, to label the protein code, another with radioactive P32 to label the DNA. Infection, radioactively labeled phages were allowed to infect E. coli. Now, blending, the mixture was agitated to separate the viral protein coats from what? The protein coat and the bacterial cells were completely separated with the help of blending. Now, centrifugation, the solution was centrifuged. In the pellet, they contained bacterial cells. The pellet which was there, it contained the bacterial cells, and the supernatant which was there, it contained the viral protein coats. Observation, in S35 labeled phages, radioactivity was found in the supernatant. That is, proteins did not enter the bacteria, it was sure. Now, in P32 labeled phages, radioactivity was found in the pellet, my dear children, in the cells. So, DNA entered the bacteria. Done. And with this, we cover Hershey and Chase experiment. This was the last experiment to prove that yes, DNA is the genetic material. Now, going ahead, okay, so there are two things for you guys. Now, you know KCET, there's an exam which would lead you get into the top colleges of Karnataka. Okay, so basically, you have two ways. to get into it. One is by taking Abhyas KCET test series, which is completely online, my dear children. Another one is Lakshya KCET crash course for those people who are in Bangalore, who think that we need teachers, we want to sit with them and study. So it's for you guys, my dear children. So you have two options, and both of them would be perfectly good for you if you are someone who is very focused about getting into great colleges of Karnataka. You get it? Now for Abhyas KCET test series, it is available for rupees 1499. And what are you going to get over here? You are going to get 35 full syllabus and chapter wise tests. There will be detailed video solution and analysis. There will be chapter wise one shot revision videos and chapter wise insight for last 5 years of KCET papers. I hope this is very much clear to you. Okay, now Lakshya KCET crash course, again there will be daily practice tests, one-on-one doubt session, guided study hours, PYQ focus, full syllabus coverage, everything. But as I said, this is a 21-day course. It's a residential or day scholar course that you have to come over here and study. Either way you can choose, and if you want any more details, children, the link will be provided in the description box. You just have to click on that. If you think you want to go ahead with KCET, if you don't think you want to go ahead with KCET, then drop it. But if you're someone who wants to get into engineering background and things like that, then I'd say definitely you should be trying this out. Okay? Chalo. Now the next chapter is evolution. So as you can see, 1, 2 and 3 marker each will be fine, but no 5 marker question is there in evolution. So it's okay. 5 marker questions are not there over here. Human health and diseases, it's of 11 marks. There will be 1 or 2 5 marker questions. So we have to be prepared for that. Now the first question, name the causative agents of the following diseases. So we should be knowing the diseases. Malaria, who causes it? Plasmodium. Who causes it, dear children? Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae. There are 3 things. You can write 3 names. Okay. Now filariasis. Filariasis is caused by Wuchereria bancrofti or Wuchereria malai. It is what? Basically an Aschelminthes. It is a nematode. It is an Aschelminthes. You might have studied about Platyhelminthes, Aschelminthes and things like that. So with love, we call Aschelminthes as roundworms, my dear children, are basically nematodes. But filariasis is caused by one such, and you call it as Wuchereria bancrofti or Wuchereria malai. There's 2 species. Now Escheriasis, another Aschelminthes. Escheriasis is caused by Ascaris lumbricoides, which is again what? A Aschelminth. Okay, Ascaris is the thing that you can write. You don't have to write lumbricoids and thing because it's not given in NCERT. At least you write Ascaris. Now amoebiasis is caused by ant amoeba histolytica, which is a protozoan. So malaria and amoebiasis, both of them are protozoan diseases, my dear children. So it is caused by ant amoeba histolytica. I will be showing you the spelling as well. Pneumonia, in your book, it is bacterial disease, my dear children. It is caused by Streptococcus pneumoniae or Haemophilus influenzae. Streptococcus pneumoniae or Haemophilus influenzae. So how do you write? Everything is being put up over here. So Plasmodium, P. vivax, P. malaria, P. falciparum is there. It is the most malignant form of malaria is being caused by this guy. Okay. Now filariasis by Wuchereria, Wuchereria bancrofti, Wuchereria malai. Ascariasis by Ascaris, amoebiasis by ant amoeba histolytica, pneumonia by Streptococcus pneumoniae and Haemophilus influenzae. Works? Okay. Next, what are lymphoid organs? Write the functions of the following in response to immune system or in our body. So bone marrow, thymus, spleen and lymph nodes are given over here. First thing, what are lymphoid organs, my dear children? These are the organs where basically the blood cells and everything would be formed. So you have 2 categories. One is primary lymphoid organ and the other one is secondary lymphoid organ. So primary is the place where you have, bone marrow is there. Bone marrow is there. What does the bone marrow contains? It contains stem cells, hematopoietic stem cells, which are basically blood cells. Okay, they are going to divide into not blood cells. They are hematopoietic stem cells. That means they are the ones that will be giving rise to different type of blood cells, such as RBC, such as WBC, and things like that. So there will be RBC, WBC platelets all being formed in the bone marrow itself. Now RBC fine, WBC fine. Okay, in WBC also you have different types: neutrophils, basophils, that phils, this phils. So many things are there. Now in that WBC you have B lymphocytes and T lymphocytes. B lymphocytes and T lymphocytes. So B lymphocytes basically mature over here in the bone marrow itself. Bone marrow itself. But T lymphocytes, they move to the next primary lymphoid organ, which is thymus. In the thymus they would be maturing. And I'll tell you one more thing. So they are produced there. So they are produced. Bone marrow is a place where all of them are produced. Now from bone marrow, T lymphocytes will be going to the thymus for maturation. B lymphocytes will be maturing there itself in the bone marrow. Now after that, they are also going to be antigen sensitive. Okay, they are going to differentiate into what? They are going to mature into what? Antigen sensitive lymphocytes. As in, they are the ones which would be able to detect the antigens and basically work on it. So both these functions have been done by the primary lymphoid organs with this bone marrow and thymus both. Okay, now from there they are going to actually do the work. Okay, they are actually going to face the antigens, not in the primary lymphoid organs, in the secondary lymphoid organs. They are going to face what? The antigen, the foreign particles, and they are going to deal with it. They are going to do dishum in the secondary lymphoid organs. So, the primary lymphoid organs of bone marrow and thymus, where immature lymphocytes differentiate into antigen sensitive lymphocytes. Cool? So, they are produced and they differentiate there. Now, after maturation of lymphocytes, they migrate to secondary lymphoid organs such as spleen, lymph node, tonsils, Peyer's patch of small intestine and appendix. The secondary lymphoid organs provide the site for interaction of lymphocyte with antigen, where they proliferate to become what? The effector cells. So, this is where, okay, in the secondary lymphoid organ, this is where they would proliferate, they will increase their number to become what? The effector cells. They are going to kill all the cells over there. Okay, in the spleen, lymph nodes and what do you call tonsils, Peyer's patch and everything. You know, tonsils, Peyer's patch, everything is a part of MALT. What is MALT? Mucosa associated lymphoid tissue. So, we have different layers. Okay, we have mucosa, the innermost layer, then we have submucosa and things like that. Okay, so innermost layer mucosa, in that whatever associated lymphoid tissues are there, okay, those are known as MALT, mucosa associated lymphoid tissues. So, you have tonsils, Peyer's patch, everything which is like that. Peyer's patch is found in the intestine, tonsils are found over here. Got it? And then you have spleen and lymph nodes. Now we are asked about the functions of each. So, bone marrow, the primary site to produce hematopoietic stem cells that later give rise to red blood cells, white blood cells and platelets. And also this is where the B lymphocytes mature. This is where the B lymphocytes are gonna mature. Now over here, thymus, an organ where T lymphocytes mature, my dear children. Okay, I told you. And thymus is active in childhood and in early adolescence, but will be shrinking after puberty. Slowly it will shrink down. Now spleen is what? An organ that filters blood. So what does spleen have? So spleen is basically the thing above your kidney, above your stomach, my dear children. So basically what happens in over here, whatever RBC comes over here, we call it spleen as RBC's graveyard of RBC. So what old and worn out RBCs come, no, they are being killed over here. They have been broken down. Whatever iron is there is being taken up and rest of the things is being degraded. Now here is the same place where you have so many WBCs, that is lymphocytes. What do? They also trap pathogens. So whenever the blood comes, they trap pathogens in there. So spleen also is an area that is acting as a secondary lymphoid organ, because this is where the WBCs will be fighting against all the things. So it's an organ that filters blood, removing old or damaged red blood cells, and also helps in immune response by storing white blood cells and acting as a site where immune cells can detect and respond to pathogens. So two functions have been done. Now lymph nodes, if you are talking about lymph nodes. So what is the function of lymphatic vessels? You know, in the red blood cells, in normal cells of our body, okay, there are normal cells. Now to reach to these normal cells, the blood vessels which are there, they will break down into small capillaries. They will break down into small capillaries. Now the problem with this capillary is that they have small pores through which things can easily move out. So there will be a lot of plasma, there will be a lot of cells that will be moving out, my dear children. Okay, there will be a lot of things that will be moving out. Okay, plasma, certain cells and things like that. So basically, what exactly happens over here when it moves out? When it moves out, what exactly is happening over here? It will cause inflammation, and we do not want extra fluid to gather up over here. So we want another lymphatic vessel, another vessel to carry this thing, and that vessel is lymphatic vessel. They will be carrying all the extra fluids and everything and give it back to the vein and everything. So while doing so, okay, this lymphatic vessels which are there, they have certain regions which are known as lymph nodes, my dear children, which are richly supplied with WBCs. So whatever comes through here, if there is pathogen, they are going to kill it. So that is a function of lymph nodes. They are small bean-shaped structures that filter lymph, okay. They trap the foreign particles such as pathogen and are key sites for immune cell activation as well. So when you go to doctor sometimes touches here to check if your lymph nodes are swollen. If they are swollen, that means you are probably facing something in your body. There is some trouble happening in your body, infection or something like that. You get it? So that's your body's way of showing things. Yes. So these are the things that you definitely need to write down. Apart from that, you have tonsils and Peyer's patch as well. Now going ahead, explain different techniques which help in cancer detection and diagnosis. So basically, detection and diagnosis is a very what's important topic. So over here, whenever someone has cancer, you know that particular cell, that group of cells are being taken up. It's been cut and been taken up. This process is known as biopsy, where it is being cut and being removed. A small part of it is being taken up for treatment. Now, not for treatment, for basically pathological studies. And what do you call this pathological studies? As histopathology. You call this as histopathology. So basically, after biopsy, after cutting down a few cells or tissue, basically a group of tissue, and then you are going to do histopathological study. That is first technique for detection as well as diagnosis. Apart from that, what do you have? So biopsy and histopathology. In biopsy, a piece of suspected tissue cut into thin section is stained and examined under microscope, histopathological studies. Now, imaging techniques are there. There is computed tomography that is going to use X-ray, that is CT scan. You say CT scan, right? It uses X-ray to generate a three-dimensional image of internals of an object, or basically of your organs and things like that. So, all your internal things are being seen with the help of a CT scan, which uses X-ray, my dear children. Now, there is MRI, which is a safer option. Okay, so MRI uses strong magnetic fields and non-ionizing radiation to accurately detect pathological and physiological changes in the living tissue over there. So computer tomography uses X-ray, please remember that, and it gives you three-dimensional image of your internal organs. Now MRI uses magnetic resonance, okay, magnetic fields, okay, magnetic resonance, MRI is indicator. Okay, so basically MRI uses strong magnetic fields and non-ionizing radiation to accurately detect all the things which are in there and to find out any changes in the living tissue. Okay. Okay. Now what we have? Magnetic resonance imaging, that is MRI. Now antibodies against cancer specific antigens that are also used for detection of a certain cancer. So you know, in our body, every time when we have something, they will be releasing certain antibodies. We get it? So we can check those antibodies in our body. For example, if someone has cancer and we know that in response to this particular cancer, certain antibodies, this type of antibodies are going to be released. Now, if I am checking my blood and I find this type of antibodies in a elevated amount in my body, that means what? Probably I might also be having this particular condition. You get it? So antibodies against cancer specific antigens are also used to detect certain cancers. Now techniques of molecular biology can be applied to detect genes in individuals with inherited susceptibility to certain cancers. So, for example, breast cancer. Breast cancer is found in generation after generation. So basically there are techniques in molecular biology that would be helping in understanding cancer in certain individuals where it is seen that it is inherited. Okay, the inheritance pattern is very strong. So yes, so these are the things that are used for cancer detection and diagnosis. Now going ahead, some drug bottles had their name labels missing in a drugstore of a hospital. The staff needs to identify the drugs with their actions still written on them, analyze their actions listed below and identify name of each drug and also the source of each of them. So we have to find each of the drugs and we have to find their source also. So drugs name and function is written, we have to just find it. So it is used by doctors as sedative and painkiller. So there is only one drug which is being mentioned in your NCERT, and that is morphine, which is going to act like a painkiller as well as a sedative. That means that person will be able to sleep as well as it is going to kill the pain. Now this, what is the source for morphine, my dear children? Source for morphine is Papaver somniferum, which is opium poppy. Okay. Now second thing, it helps patient to cope with insomnia and depression. So again, in your NCERT you have two drugs, that is barbiturates and amphetamines. Fine. Just a second, okay. Akash, you wanna do something?
Biology 5 marker.
I'm doing it.
This is the video?
Yes.
Where is this pause button?
I don't know.
Okay.
Like 1/2 is done. You want to take it?
No.
That is of 2 hours.
You know, no, now it's storing in server.
Yeah? Yeah.
Don't move. Search there.
But
in previous folder. It won't be there in previous folder where it used to save.
So the older one, where do you find it?
Older one will be there only. Now what is recording will be saved in server.
Okay, fine. So you don't want to take the older one? No.
I thought it was in that folder.
Okay.
Nothing.
Okay, take care. Okay, so it helps patients to cope with insomnia and depression, my dear children. So over here, it is barbiturates and amphetamines, and the source is obviously not given in your NCERT, so you do not have to be worried about them. Okay, now next, increases blood pressure and heart rate of consumer. Blood pressure and heart rate of consumer, this is nicotine, my dear children. This is nicotine which is there, and act as depressant and slows down body function. That is, it is acting as depressant and it is slowing down body function. This has to be heroin, my dear children. That is the only depressant mentioned, and again the source is papaver somniferum. Now, affects cardiovascular system of your body. So there is only one thing that affects the cardiovascular system of your body, and that is cannabinoids. Cannabinoids obtained from cannabis sativa. Obtained from cannabis sativa. Okay, example of cannabinoids, marijuana, ganja, everything. So you can write either of them. So if you do not know the functions, my dear children, you would not be able to write it, which is why you should know the function really well. I have written all the answers for the question. Works? Chalo. Next thing. Describe the measures taken to control alcohol and drug abuse in adolescents. So these are very simple things, okay. So avoiding undue peer pressure, that would obviously do good. Education and counseling about how wood is bad. Seeking help from parents and peers, you should be definitely doing that, okay. And looking for danger signs and seeking professional and medical help. So these are the measures taken for controlling alcohol and drug abuse in adolescents. Just go through it. If I keep on explaining, the time would be really long. I do not want that to happen. Yes, it's already 38 minutes. So yes. Next thing, give the schematic representation of replication of retroviruses. So what exactly happens over here? This is schematic representation, so you have to represent like this, my dear children. So first is viral core. So over here, you know, the virus has capsid and everything, protein capsid covering, and then it will be having its RNA thingy. It has RNA, we have DNA, okay. Along with RNA, there will be certain enzymes also over here. There will be certain enzymes that are pre-prepared over here in the viral core. Now, this virus is going to affect a cell. Suppose this is a macrophage. Inside the body of human beings, the first cell that goes and affect is a macrophage. Okay. When it comes and affects, it is going to send its viral RNA. Viral RNA is introduced into the cell, as you can see over here. And as I said, they have certain enzymes also, and the first enzyme is reverse transcriptase. What does reverse transcriptase do? It reverses the transcription. You know, usually DNA is the one that comes first. The DNA is transcribed to form RNA. That's how it works. But over here, RNA is there, and now you're going to make a DNA out of it, which is why it is known as reverse transcriptase. So reverse transcriptase enzyme will be making a DNA out of it, viral DNA, and this viral DNA is going to incorporate over here. It is going to what? Incorporate over here in our genome. So viral DNA incorporates into host genome, and now it will take all the usage of it, as in the replication, everything, duplication, everything will be happening. So there will be transcription also will be happening. So there will be new viral RNA that is produced by the infected cell, my dear children, and there will be viruses. There's so much RNA is there, and the proteins are also being produced. From the mRNA, you get the proteins also. So they will be getting the capsid and everything ready. Now they are going to get released, and they are going to infect the other type of cells as well. You get it? So this is how things work. So new viruses can infect other cells. It is usually the T lymphocytes, T helper lymphocyte, that it is going to be affected because of this. So first macrophage will act like a HIV factory. It will release more and more of them, and then later these particular HIV viruses, my dear children, they are going to affect the T helper lymphocytes, and basically when T helper lymphocytes is affected, all the other immunological things in our body goes really down. Cool? Chalo. And with that, we finish that particular topic as well, that is human health and diseases, my dear children, and we are going ahead with microbes in human welfare, which is a 7-marker chapter. So either 1 5-marker or none would be coming from this particular chapter. So for 1 5-marker, we are looking forward to this particular chapter. Okay, so let's hope that it comes, because we are fully prepared. Aren't we? Yes, right. So with respect to microbial products, its source and uses, identify A, B and C in the following table. So cyclosporin A. What is the source of cyclosporin A, my dear children? It is being produced from a fungus, that is Trichoderma polysporum. And what does it do? It is an immunosuppressant, as in you want your immune system to suppress for some time, because if it is active, it is going to. Okay, I'll give an example and I'll tell. So I am a person who have my kidney damaged. So I want kidney transplantation to happen. I took the kidney from one of my relatives. Now I have it in my body. Now my body feels this is foreign and goes react with it and tries to damage it. But is it beneficial for me? No. At this point of time, I want to suppress my immune system. That is why I take immunosuppressants. Okay. So where do you get cyclosporin A from? You get it from Trichoderma polysporum, which is what? A fungus. Now Monascus purpureus is a source. It is a blood cholesterol lowering agent. So what do you get from Monascus purpureus? You get statin, my dear children. Statin stabilizes cholesterol. You can remember like that. Now you have streptokinase, you have streptococcus. Streptokinase is an enzyme that you get from streptococcus, my dear children. And what is streptococcus? It's a bacteria. It's a bacteria. So what is the use of streptokinase? It is the one that acts as clot buster. So you know clots might be formed in our body and it has to be removed. Otherwise the blood flow would be clogging. So we do not want that to happen. So it acts like a clot buster. Over here streptokinase is going to act like a clot buster. Cool. Now these are the three things that you have to fill over here. I will just say about BOD and flocks and then I would be going ahead, or probably about BOD and flocks I will be talking later when I talk about sewage treatment, because anyway sewage treatment is over here. So I'll talk then later. Okay, this question will be discussed later. I will be showing you just the answer for this. So cyclosporin, Trichoderma polysporum is the answer. Okay, statin, Monascus purpureus, blood cholesterol, and over here streptokinase, streptococcus, clot buster is the thing. Cool. Chalo. Next thing, explain a typical biogas plant with a neatly labeled diagram. So how do you explain biogas plant? So over here, this place is where you're going to put dung and everything, dung and water, and you're going to mix it. Okay, the animal dung, and then this will slowly get into this particular tank. You know, here there is an anaerobic situation. That means there is no oxygen, and when there is no oxygen, what will happen? The anaerobic bacteria will flourish. They will flourish. So in the cow dung, what do you have? You have methanogens, certain type of bacteria that is responsible for production of methane when there is an anaerobic environment. So these methanobacteria or methanogens, basically they are going to reach over here. They are going to do anaerobic respiration, and they are going to release lots and lots of what? Methane, H2S, and also carbon dioxide. So methane, H2S and carbon dioxide has been removed over here with the help of this gas outlet. This can be used as a biogas. Okay, you got it? And it can be used in kitchens and all, or for providing electricity. Now whatever is left behind can be taken up over here in this particular tank. From here it can be used like fertilizers and manure as well. So this is a biogas plant, my dear children. You have to explain like this. So biogas plant consists of a concrete tank that is 10 to 15 feet deep, in which bio waste are collected and slurry of dung is fed. A floating cover is placed over the slurry, which keeps on rising as the gas is produced in the tank due to microbial activity, that is methanogens. The biogas plant has an outlet which is connected to a pipe to supply biogas to nearby houses. Used slurry is removed through another outlet and may be used as fertilizers. Now the biogas thus produced is used for cooking and lighting, and the technology of biogas production was developed in India, very important MCQ question, by Indian Agricultural Research Institute, IARI, and also Khadi and Village Industries Commission, KVIC. Okay, so this is the answer that you have to write. Next, explain the stages of sewage treatment. Yes. Chalo, let's start. So in sewage treatment, my dear children, you have to remember two stages. One is primary treatment and the other one is secondary treatment. So what do you do in primary treatment? In primary treatment, you have two things. One is filtration and the other one is sedimentation. As in, the things that are going to float, you can just easily pick them and throw it out, or basically remove it out, right? So this is known as filtration. So first is filtration. And the other one which is there, it is known as sedimentation. What do you mean by sedimentation? Whatever is left behind is being sedimenting towards down. Whatever is there floating, you take it out. Whatever is heavy, it will be sedimenting down. You can remove it as such. Now what are you left with? You are left with something that is known as primary effluent. You are left with something that is known as primary effluent, my dear children. Now you are going to use this for secondary treatment. For what? Secondary treatment. What happens in secondary treatment? Listen to me very carefully. Okay, so in secondary treatment, things are being put in aeration tank. Aeration tank. And what happens in aeration tank, my dear children? There will be lot of air, and if there are lot of air, there will be lot of aerobic bacteria. There will be lot of what? Aerobic bacteria. Agreed? Agreed? And this primary effluent which is being passed down, it will obviously have something that is known as organic matter, which has to be broken down. okay, which has to be broken down. So what happens is that this aerobic bacteria is going to act on the organic matter. This aerobic bacteria needs oxygen. Needs what? Oxygen. And this need of oxygen is known as biochemical oxygen demand, BOD. There was a question like BOD. What is BOD? Here you will get the answer. What is BOD? BOD is the amount of oxygen required by microorganisms for breaking down organic matter in 1 liter of water. So organic matter is there. For breaking it down, you need aerobic bacteria. Aerobic bacteria in turn needs what? Oxygen, my dear children. So in the presence of oxygen, they are going to break it down in the aeration tank. Got it? In the aeration tank. Now, one very important thing is that it will keep on going until the amount of BOD would be reduced. What do you mean by that? The amount of oxygen needed will be reduced by, until then it will keep on going. So if suppose organic matter ends, okay, it is broken down completely, the aerobic bacteria would not need more amount of oxygen now, no, because they have broken down. So the amount of BOD, biochemical oxygen demand, will also reduce. So at this point of time, until this point of time, the whole thing is being continued. And let me tell you one more thing. So while it does so, no, the aerobic bacteria will be combining with certain fungal filaments and stuff like that, and together forming a mesh-like thing which is known as flux. So flux is nothing but a mesh-like thing which has what? Aerobic bacteria for fungus filaments plus fungal filaments plus what? Extracellular debris which is there. Okay, now the thing is being cleared. Okay, now what do? You're going to pass it down into a settling tank again. What will happen? All this mesh-like thingy which is there, it is going to sediment right down over here. This is known as activated sludge. This is known as activated sludge. And now what do you have over here? The effluent, the treated effluent. This is safe now. It can be transferred anywhere in the river, stream, whatever is there. Now activated sludge from here, that means it contains aerobic bacteria and everything. No aerobic. I can take a little bit of it and put it down in this particular aeration tank. For the next treatment to happen, you need certain bacteria. So it is going to act like inoculum. So it is going to act like inoculum, my dear children. Now that has been done. Rest of the thing is been there. So you have removed this, now you do not have this now, okay. You have removed this into water, rivers and streams and things like that. You gave some part of it to as an inoculum also. Now you are left with some amount of this. Now you are going to put it inside. You are going to put it inside an aerobic sludge digester. Okay, you are going to put inside an aerobic sludge digester. What happens in an aerobic sludge digester, my dear children? There is anaerobic situation. That means there is very less amount of oxygen. There is what? Very less amount of oxygen. There is no oxygen actually. Anaerobic as in there is no oxygen at all. Okay, sorry, I was zoned out. Yeah. So there is no oxygen up over here. So what will happen? The anaerobic bacteria, my dear children, the anaerobic bacteria will start acting on the aerobic bacteria, the fungal filaments and everything, and they are going to release what? The biogas, that is H2S, methane as well as CO2. Now this biogas can be used. So basically with the help of primary treatment, you are treating everything over there. Okay, the organic matter is completely being broken down. Okay, whatever you get from these things, that is also being treated down for biogas treatment and for biogas production and things like that, and rest of the treated thing, water is being removed to rivers and things like that. Got it? So over here, how do you write it? So primary treatment involves physical removal of particles large and small from the sewage. So initially floating debris is removed through sequential filtration. The grit are removed by sedimentation. All solid that settle from the primary sludge and superintendent forms the effluent. The effluent are from the primary settling tank taken to secondary treatment. Now the primary effluent is passed into large aeration tanks. This allows vigorous growth of useful aerobic bacteria into flocks, my dear children. The growth of microbes consumes a major part of organic matter in the effluent. This significantly reduces the BOD, biochemical oxygen demand. BOD refers to the amount of oxygen required to oxidize total organic matter by bacteria present in 1 liter of water. Now, it is a measure of organic matter present in the water. Greater the BOD of water, that means what? Greater oxygen requirement is there. That means more amount of organic matter is there. That means more pollution is there. Get it? So, once the BOD of sewage reduces significantly, the effluent is then passed into settling tank. Small part of where the bacterial flocks are allowed to sediment. This sediment is known as activated sludge. Small part of this sludge is pumped back into aeration tank to serve as inoculum. The remaining sludge is passed to anaerobic sludge digester. In anaerobic sludge digester, there is other kind of bacteria which grows anaerobically, digest the bacteria and fungi in the sludge. During digestion, bacteria produces biogas, and that's all. Got it? So that's what I said. Okay, that's the exact thing that I said. Now, if you want the answer for flux and this, so BOD is the amount of oxygen required by microorganisms to decompose the organic matter present in a given water sample, that is 1 liter of water. That is what you say. Flux are clumps or clusters of fungal filaments, bacterial cells and other materials that are held together by extracellular substances produced by microorganisms. These aggregates are involved in biodegradation of organic matter during processes like wastewater treatment. Done? Okay. Now, we have biotechnology principles and processes, again giving you one 5-marker question. So, we have to look forward to that, and let me just see how much questions are left. So, would we be able to do it really fast? The answer to this question is yes, we would be able to do it really fast. So, given below are diagrams of plasmid A and B, my dear children. This is A, this is B. Okay, observe meticulously and answer the question that follows. Which plasmid is or are would you select for cloning and why? So, I have 2 plasmids. I have 2 plasmids, my dear children, this and this. So, which one will I actually use for cloning and why? I can use both of them. Why, you may ask? So, see, I will start with this plasmid number B. So, LAG-Z gene is there. Okay, LAG-Z gene is the one that would be giving rise to or basically producing an enzyme, that is beta galactosidase. And what does this beta galactosidase do? It is going to break some things down. Okay, so in this medium, whenever you have whenever you have LAG-Z and you are going to treat it with some coloring agent, what happens is that the beta galactosidase enzyme which is there, it is going to break down this coloring agent, giving rise to blue color colonies. The color will be blue in color, not brew, it is blue in color. So basically, now if I am inserting something else instead of LAG-Z, over here when I insert something else, now beta galactosidase would not be produced. Now do you think blue color will be produced? No, blue color will not be produced. What will I see? White color colonies. So it is very easy for me to understand whether this particular plasmid has taken up the gene or not, which is why I would be taking this. Now on the other hand, if we talk about plasmid A, over here it has ampicillin resistant gene and tetracycline resistant gene. That means now it has genes for both these antibiotics. If I try to grow in ampicillin, it will still grow. If I try to grow in tetracycline, it will still grow. But now what I am doing, I am going to insert a gene of my interest into tetracycline resistant gene over here. Now is it tetracycline resistant anymore? No, my dear children. So again, if I have to find out whether it has taken up this particular gene or not, what do? At first, I would be making it grow in ampicillin resistant gene condition. That means I will be putting in ampicillin and see whether it is growing or not. I will be finding that it is growing. All of them basically would be growing. Now the ones that would be having some other gene over here, okay, the ones that would be having some other gene over here instead of tetracycline would not be able to grow in tetracycline resistant medium. Okay, or basically in tetracycline medium. If I am putting tetracycline in a medium, suppose over here I am putting tetracycline and I am making the plasmids grow. Will this particular plasmid grow where I have put something else instead of, so this has the gene for tetracycline resistant, this is my gene of interest. So here it is not resistant, here this is resistant. So this will be able to grow, but this would not be able to grow, my dear children. That means it has taken up my gene. So I can use both of them because in both of the cases I am able to understand whether the plasmid have taken things up. And now I can insert this particular plasmid into the organism that I am looking forward for. We get it? So that is the thing. So how do I frame? Both plasmids allow for cloning via insertional inactivation, but plasmid B might be preferred for ease of screening using blue-white method. So it will be very easy for me to see this rather than that. Okay, because here the color changes won't be happening, it will be giving me white colony, so it is easy for me to understand that. Now, what is insertional inactivation? You might have seen that, okay, in the answer itself, but what is insertional inactivation? Basically, when I am inserting some gene and because of it, the older gene is getting inactivated, that is known as insertional inactivation. Over here, instead of LAG-Z, I am putting my gene of interest. So, isn't this particular gene getting inactivated? Yes. So, this is insertional inactivation, my dear children. Got it. So, insertional inactivation is a process where insertion of foreign DNA into a marker gene, that is tetracycline in plasmid A or LAG-Z in plasmid B, disrupts its normal function. This disruption results in detectable phenotype such as loss of tetracycline resistance in plasmid A, loss of beta galactosidase activity in plasmid B. Cool. Now, talking about the next thing, will the number of culture plating you should make is same or different to select recombinant if insertional inactivation is possible? So, over here, you know culture plating. So over here, listen, we have to read this first, otherwise you might not understand. So plasmid B, will start with that. It contains the LAGZ gene which allows for insertional inactivation using blue-white screening method. Recombinant disrupts the LAGZ gene leading to white colonies on X-gal media. Non-recombinants produce beta-carotidase turning blue on the same medium. So over here, what do? We have a medium, okay? We have a medium over here. In that, basically, there are bacteria with this particular plasmid, okay, that has my gene of interest. Okay. Similarly, I have another group of bacteria with plasmid which does not have my gene of interest, but it has LAGZ gene. Now I am putting my coloring thing, that is X-gal. When I am putting X-gal over here, this guy will be producing beta-galactosidase and it will be giving blue colonies over here, blue coloration over here. Whereas this is not going to produce any beta-galactosidase, hence it will be still white in color. So in one particular medium itself, I do not need two culture plates or things like that. In one particular plate, the white colonies can be separated from the blue colonies easily. The white colonies can be separated from the blue colonies. So the white ones can be taken up for further process. The blue ones, I do not need them because they are not taking up my particular thing right over here. Got it? Got it? So these are going to grow, these are going to give me white colonies, these are going to give me blue colonies in the same plate. So one culture plating is required. So will the number of culture plating you should make is same or different to select recombinant? If insertional inactivation is possible, this will be same is enough. You don't need much. Now, on the other hand, if we talk about plasmid A, there what do? You have one plate, okay, where you are going to grow E. coli. Suppose this is an E. coli. Okay, both of them will be having ampicillin resistant gene, but one would be tetracycline resistant, other one will not be. The white one is not, okay. So first plating is done in ampicillin medium. So both of them will be surviving. That means both of them has taken up the plasmid. In E. coli, both of the E. coli has taken up the plasmid. Cool. Now I have to. So they both are transformed. So they both are transformed. I call these cells as transformed cells because they have taken up the plasmid. Initially they didn't have this plasmid, I put this plasmid in them and they have taken it up. Now I have the next medium. This is for selecting recombinants, the one that has my gene of interest, okay, one that have my gene of interest. So over here, my dear children, what do I do? What do I do is that again I have two things. Over here my gene of interest is there, and over here I have the tetracycline resistant. Now what do? I put in tetracycline medium. I will be saying that this is able to survive, this is not able to survive. That means this is the one that is recombinant because it is not able to grow in number. It is the one that is not able to grow in number, that is why this is the one that is recombinant. But this is not the recombinant, my dear children. This has the tetracycline resistant gene. So this is the next plating that I require. Otherwise I would not be able to understand. So here you need two platings. So requires two-step plating, plate on ampicillin containing medium to ensure plasmid uptake, replication on tetracycline containing medium to identify the recombinants which will not grow on tetracycline. So in this way I am able to identify that this is the one that is not able to grow, that means this has taken up my gene. Okay, got it? Cool. Now study the diagram given below and answer the question that follows. What is EcoR1? You know, EcoR1 is a restriction enzyme. Restriction enzymes are the one that acts as molecular scissors. They are going to cut the DNA, my dear children. So they have a specific site where they cut. They don't do random cuts. Okay, so for EcoR1 is being derived from E. coli strain. Okay, and this is going to cut at GAA TTC strand. So 5' to 3'. This particular unit it is going to cut. This is the sequence that it finds, and it will be cutting between G and A, my dear children. It will be cutting between G and A. Now, how is action of exonuclease different from that of endonuclease? So, in restriction enzyme, there are two types. One is exonuclease and the other one is endonuclease. Exonucleases are the one that will cut DNA from the ends. Endonucleases, for example, EcoR1, it will be cutting from the middle. Anyway, in the middle of the DNA also it can go and cut, which is why it is known as endonuclease. Got it? So here is the answer. So first thing, EcoR1 is a restriction enzyme that recognizes specific DNA sequence at 5' GAA-TTC and cleaves between G and A. It is derived from Escherichia coli strain RY13. That is why R has come, eco R from RY strain, and 1 means it is numbered according to discovery. Now exonuclease removes nucleotides from the ends of the DNA or RNA. Okay, loss of, okay, don't know why it is there. Now next thing, endonuclease cuts within the DNA or RNA strand, breaking internal phosphodiester bonds. Now talking about this one, how are sticky ends formed on a DNA strand? Why are they so cold? So why are they known as sticky ends and how are they formed? So over here you see, okay, so this is the vector DNA and this is the foreign DNA that I am having. Okay, this is the vector DNA and this is the foreign DNA that I am having. I am going to put this over here. So I have to mix them basically. So what do? I am going to cut from here. GAA-TTC site, no, I will be cutting with restriction enzyme, that is eco R1. It will make a cut over here. Similarly, it is going to make a cut over here. Same enzyme is used to make a cut over here also. GAA-TTC, it finds, it makes a cut over there and there. So what do you find? Basically, you see a L-shaped thing right over here. From here it is cut and from here also it is being cut. Now you have, what say, single stranded region which is not complementary, which is, he does not have any complementary thing over here because it is being removed, my dear children. Here also it is being removed. Now you only think, I want to join both of this vector DNA and the foreign DNA. What do? I just make it come together. So these are known as sticky ends because it does not have a complementary base pair, but when it finds a complementary base pair, it will vastly stick to each other. Okay, and then I can basically use DNA ligase to properly make it stick to each other. So that is the use of sticky ends which are there over here. So how to explain it? Okay, eco R1 cuts DNA at recognition site in a staggered manner, as in a like this manner. This is staggered manner. It is not proper manner. Okay, it is not a good finish. Get it? The staggered manner. Now what will happen? So it will leave overhanging single stranded ends which will like this. Now these overhangs can easily form hydrogen bonds with complementary sequences, allowing them to stick to each other DNA fragments with matching ends. This property is essential for DNA fragments to form recombinant DNA technology. So you don't need to write all this thing over here, you just have to explain this. You can either show this, that will be more than enough. Okay, this will be more than enough to show. Cool. Now, write 3 vector free techniques that can be utilized to transfer recombinant DNA into a ready host cell. 3 vector free techniques. You don't need vector things to basically an organism or things to basically transfer DNA into it. Okay, what are the vector free techniques? And also, agrobacterium tumefaciens acts as a natural vector for cloning genes in plants. Substantiate, you have to explain that. So let's deal with the first part of the question. So first part of the question, I told you vector free techniques you need, right? So first thing is electroporation. What do? A high voltage electric pulse is created which will be making temporary pores, my dear children. So it allows the DNA which is there to enter the particular thing, the cell. Now there is micro injection. Now micro injection, please remember, it is limited to animal cells. So here DNA is directly injected into the nucleus with the help of injection. DNA which is there, it is put inside the nucleus of the host cell using what? Fine glass micropipette. Now there is gene gun which is used in the case of plants. We know plants have cell wall. So obviously you cannot use micro injection, you need gun. So gene gun, it is also known as biolistic. It is where the DNA is coated. Okay, DNA is coated around microscopic particles of gold and tungsten and then it is shooted in. So what happens basically when this goes in, it is passing the DNA as well. So these are the 3 vector free techniques that you can remember. Now talking about agrobacterium tumefaciens, it is basically a bacteria that causes tumor in the case of plants. How does it cause tumor? Basically this particular bacteria has a plasmid which is known as TI plasmid, my dear children. Tumor inducing plasmid. Why? Because it has a segment of DNA which is known as tDNA. tDNA stands for transfer DNA. So basically when this particular bacteria affects the plant, my dear children, it is going to transfer this segment of the DNA into the plant's body. And when it transfers, so what will happen? It is going to cause what? Tumor in the case of plant. Okay. Now I am going to use this agrobacterium tumefaciens as a natural vector for putting genes into the plants. How can I do that? I can remove this tDNA and I can put the gene of interest over here. I can put the gene of interest over here. Now when I put the gene of interest over here, what will happen? What will happen? Basically Again, the TI plasmid will try to go and infect. This time it is passing my gene of interest rather than the transfer DNA. So that is the use of Agrobacterium tumefaciens. So how do you write it? Agrobacterium tumefaciens contain a TI plasmid, okay, that can transfer a segment of DNA. This tDNA integrates into the plant genome, my dear children. Use in biotechnology is that scientists modify the tDNA region to carry desired genes instead of tumor causing genes. The bacterium then serves as a natural delivery system to insert recombinant DNA into the plant cells, making it widely used tool for genetic engineering in plants. Done. So you just have to write this much. Okay, now the next question, that is, explain the process of denaturation, annealing and extension with respect to polymerase chain reaction. So that is PCR basically. And what happens in PCR? You have a DNA strand and you want to amplify it. So what do you do it? Basically, first thing, you break the bonds between the DNA. So that process is known as denaturation, and it happens almost around 90 to 95 degrees Celsius. Okay, so this is being separated, my dear children. Now the separated DNA which is there, onto this we are going to attach certain primers. You know, primers are required for DNA polymerase to extend it, right, to make a new copy of DNA. So basically over here we need to add the primers, and this process is known as annealing. This process is known as annealing, which again takes about 50 to 55 degrees Celsius. Okay, now after we have added the primase, okay, we have given the primase to add the primer, my dear children, and after that what do? We treat it with Taq polymerase. Taq polymerase. What is Taq polymerase? It is a form of DNA polymerase. It is form of DNA polymerase which is going to extend the whole strand up over here. Okay, and obviously for extending you need nucleotides also. So you call them DNTPs, deoxyribonucleotide triphosphates. Okay, or deoxyribose nucleotide triphosphates, either way. So it is DNTPs that you required, and also DNA polymerase, that is Taq polymerase specifically over here, which will be basically extending the DNA strands. Good, works. Okay, and this is known as extension. Again, this requires around 70 to 72 degrees Celsius. So if we see, the amount of temperature is really high, which is why we are taking DNA polymerase from an organism, that is a bacteria, which is thermostable. It is able to survive in high temperature. What do you call this? This is, the name of the bacteria is Thermus aquaticus. Name of the bacteria is Thermus aquaticus. Got it? Now we have plasmid. Define plasmid. How do you define plasmid? Plasmid is basically the extra chromosomal DNA. So suppose this is an E. coli. Now E. coli is a bacteria. E. coli bacteria will have its own DNA, my dear children. Apart from the own DNA that it has, it also has certain extra chromosomal DNA which is known as plasmid. Now it will have its own origin of replication. It will still replicate and do things like that. Cool. Now, why are restriction enzymes called molecular scissors? They are known as restriction enzymes are known as molecular scissors because basically they are going to make cuts in the DNA as well as the RNA. Those are big molecules, which is why they are known as molecular scissors of biotechnology. So how do you frame all your answers? First thing, if you talk about PCR, denaturation, it involves heating of reaction tube containing target DNA to 94 degrees Celsius. This temperature denatures and separates two strands of DNA. Now, annealing of primers. This reaction mixture is cooled down slowly to 55 degrees Celsius. The two primers pair with complementary base pairs present in the separated DNA strands. Now we are putting the primers out there, and they are going to basically be they go and tag over there on the 5' end on the 5' end, on the 3' end and the 3' end of both these strands. Now extension of the primers. The temperature of the reaction mixture is increased to 72 degrees Celsius for 1 minute. The temperature is suitable for the activity of DNA polymerase. It adds complementary deoxyribonucleotides one by one to the 3-OH of the primers. So you know, I told you, you are adding the primers on the 3' end. Why are you adding? Because the DNA polymerases in 5' to 3' direction. So you have to add it over here. From here they will keep on going. So what is happening, my dear children? It adds complementary nucleotides one by one to the 3-OH end. So here it is 3-OH end. So one by one nucleotides are added. Okay, at the end of the primers, as a result of new DNA strand is synthesized. Fine. Now plasmid is what? Extra nuclear, extra chromosomal self-replicating double-stranded circular DNA of the bacteria. Now why they are molecular scissors? That is your restriction enzyme, because they are used to cut DNA precisely. Works? Done. Now Draw a neatly labeled diagram of simple stirred tank bioreactor. It looks something like this. So you have air, sterile air inlet, okay, through which the air will be inserted, and this particular thing, okay, you have flat bladed impeller, my dear children, which will be rotating around, making sure that the air is supplied throughout, and a motor, and also acid base for pH control, steam for sterilization. Also thereafter doing all the things you need to sterilize all things. So for that you need a stream entry, and then there's a foam breaker and lot of things, and this is known as a simple stirred tank bioreactor, which is there. Okay. Now draw a neatly labeled diagram of PBR 322, which is a very important diagram. So how do you draw it? You draw it like this. So here you will be finding two ampicillin resistant gene as well as tetracycline resistant gene. On this particular gene itself, you have enzyme, restriction enzyme site. One is for BAMH1, other is for SAL1. These are restriction enzymes. They are going to cut over here. These are their restriction sites, BAMH1 sites and SAL1 site. Over here also you have PVU1 and PST1. Okay, so PVU1 and PST1. This is origin of replication onto which the DNA polymerase will come and attach and basically start replicated. Now you need replication protein also, that is ROP. Now ROP site, at the same site you also have PVU2. So here there is PVU1, here you will be having PVU2. Now apart from this, so these are found in the restriction sites, right? Either these are found in the restriction sites or basically resistant enzyme site, and over here it is found in the ROP site. Now this particular thing, EcoR1, CLAV1 and HIND3 is found over here. HIND3, it is designated like this. This is the site. So you can remember it easily. Once if I make it again, so this is your plasmid, my dear children. You will be having the ORI site, then you will be having the ROP site. In the ROP site you have PVU2 site, okay, and then you have tetracycline resistant gene, my dear children. Here you have BAMH1, BAMH1, and then you have SAL1, and here this is ampicillin resistant site, my dear children. Over here you will be having PVU1 and also PST1. Now over here you have EcoR1, you have CLAV1, and then something like this, that is HIND3. Okay, HIND3. So here there is 2, here there is 3, rest all of them are 1, 1, 1, 1. Cool. This is a very important diagram, and with that we complete biotechnology principle and processes as well. So over here biotechnology in application is of 6 marks. It does not contain 5-marker questions, so you do not have to be worried about this particular chapter. Oh, either one. Okay, just a second. Okay, so no, it's not, does not contain 1 marker. No, wait. So basically the thing is, it will be having either one 5-marker, or there will be no 5-marker. There will be 3-marker, 2-marker, but no 5-marker. Okay, so 2-marker, 3-marker we have already discussed in the 2-marker, 3-marker section. You can just go watch that. Now explain 5 benefits of creating transgenic animals. What do you mean by transgenic animals? Basically we are putting our genes into them. Okay, and that's a new form of the animals which are being formed, are transgenic animals. Now what are the uses? Basically suppose in our body there is one gene, we do not know its function. If we are basically going to insert into their particular genome, we are going to understand what is the physiological function of that particular gene. And now the same gene, if whatever gene we have, we are going to put in them, and basically then we are treating with some drugs and things like that, and we are seeing whether this is safe for our body or not. That can be done. Okay, for understanding or for treatment of diseases and things like that, that can be used. Apart from that, if we are putting one particular gene which is of our interest into them, and we are making products with the help of that, okay, we are making them produce biological products. For example, there is a cow, okay, Rossi cow, in which we put the gene for human alpha lactalbumin. So now, okay, now this what happened, basically the milk that this particular cow was producing was having this particular protein. So this is very useful for us, you get it? Or otherwise, apart from that, to treat emphysema we need alpha antitrypsin, right? So basically we are going to put that antitrypsin 1. So that particular gene which will be coding for that particular protein, my dear children, and we can utilize that particular protein now. That can be done. Apart from that, if you want to do chemical testing, okay, we are putting our genes and everything in them, and we are putting chemicals and seeing how much tolerant it can be. For vaccine safety, everything, we can be utilizing these transgenic animals. So normal physiology and development, transgenic animals can be specifically designed to allow study of how genes are regulated, how they affect the normal function of the body. If we are inserting them, we get to know that this gene has this particular thing. Study of disease. Many transgenic animals are designed to increase our understanding of how do they contribute to development of disease. They are specially made to serve as models for human diseases so that investigation of new treatment for disease can be done. Now, biological products. Transgenic animals that produce useful biological products can be created by introduction of the portion of the DNA, that is gene, which codes for particular thing, such as human protein to treat emphysema, that is alpha-1 antitrypsin, for phenylketonuria also, and cystic fibrosis also. Now, protein enrichment, that is alpha-lactalbumin that I was talking about, that can be also made to produce by other animals such as cow. Okay. Now, vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccines before they are used on humans. Now, chemical safety testing. This is known as toxicity or safety testing, where the procedure is same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. Now, since it is more sensitive, now if we are putting the chemicals, we get to know how much reaction can we expect. So, they are more sensitive, so they will be giving certain reaction. So, we will get to understand about different organisms, about different human beings also. Some animals are more sensitive to certain drugs, so we will get to know how, to what extent can things go, and you know the overall thing. So, that is the 5 benefits of transgenic animals. You should be knowing this. Now, what is RNA interference? How does RNA interference help to develop resistance in tobacco plant for nematode infection? So, there is tobacco plant which is infected by the nematode, that is Meloidocaine incognitia. Okay, it's an ask helminthus. Now, over here there is a method that is known as RNA interference that plays a major role. I'll tell you how does it work. So, basically, you know mRNA. mRNA is the one that will undergo the process of translation, my dear children, that will undergo the process of translation to form protein. Okay. Now, over here, what are you going to, this is a single stranded thing that is there. Now, suppose I am introducing a double stranded RNA over here. This is what? A double stranded RNA. Cool. Now, what I am going to do is that from this double stranded RNA, this strand is going to come and attach over here and make this mRNA double stranded. Now, if it is double stranded, do you think it can still undergo the process of translation, my dear children? No. So, this is known as RNA interference. So, it won't be able to make the protein and cause trouble in that particular organism. Now, how it can be used to develop resistance in the case of plants, my dear children? So, basically, what you have to do is, scientists first identify genes in nematode that are essential for its survival or ability to infect the plant. You understood the gene. Okay, now you're going to take the gene. Now, to deliver the silencing gene into tobacco plants, scientists use Agrobacterium tumefaciens, a bacterium that naturally transfers DNA to plant cells. So, what do? Basically, you know the gene now. Okay, now you are going to insert that particular thing. Okay, you're going to make the RNA out of it, and basically you're going to insert it into the plant with the help of Agrobacterium tumefaciens. I told you this is a vector for plants. Now, the identified nematode gene is inserted into the genome of the vector. Okay, that is into the Agrobacterium tumefaciens. This DNA sequence is then inserted into the plant genome using Agrobacterium tumefaciens infection. Now, from the Agrobacterium, it is going to the plant, which transfers the gene to the plant cells. Okay. Now, what happens? Production of dsRNA in the plant. Now, once the DNA sequence is inserted in the plant cell begins producing double-stranded RNA, my dear children. It produces double-stranded RNA responding to nematode's target gene. Now, RNA interference. When nematode feed on the plant roots, they ingest the double-stranded RNA. Plant has the double-stranded RNA. Now, when the nematode is coming to eat it, the nematode is going to get double-stranded RNA. Now, this double-stranded RNA is going to trigger RNA interference in the nematode, silencing the target gene of nematode cells. I told you it is going to form complementary pair with the mRNA and going to silence it. So, first thing what you did, you took the gene which is there from the nematode, okay, which is going to cause the problem, and then you are taking that particular thing and you're putting inside the Agrobacterium tumefaciens, and this Agrobacterium tumefaciens is going to infect the plant. Now, the plant has this particular gene. The plant will make double-stranded RNA out of the DNA. It will form a double-stranded RNA out of the DNA which was inserted. Now, this particular RNA, my dear children, now this is there in the plant, a nematode will come and eat it, the plant. What will happen? The double-stranded RNA will be getting into the nematode. Now, from this double-stranded RNA, one strand will be going and forming a pair with the mRNA, silencing it completely. So, it won't be able to produce the proteins. Okay, so it cannot properly function and hence cannot harm the plant as well. Organisms and population, my dear children, it will be carrying giving at least one 5-marker question for sure. So, what are the type of question that you can expect? So, mention the population interaction that exist among the following: Abingdon tortoise and the goats. So, you know, tortoise were there, goats were there. So, initially tortoise were there and you introduced goats over there. So, both of them were feeding on the same thing. Eventually what happened is that goat's population increased and abundant tortoise was made to go away from it. So this is example of competition. Now tiger and deer, you know, tiger is the predator, deer is the prey. So this is not parasitism, this is basically prey-predator relationship. This is prey-predator relationship. Now sea anemone and clownfish. Now this is sea anemone which is there, and this is the clownfish which is there. It cleans up everything, and sea anemone basically provides protection and everything. So both of them are helping each other, so I'll call this as mutualism. Now wasp laying egg in the fig fruit. Now wasp is an insect, it is laying an egg in the fig fruit. That means fig is providing a shelter. Now what is the use of fig fruit? What help does it get from the wasp? Basically pollination is being done with the help of the wasp, because wasp moves around here, they will be having pollen grains all around them. So this is again example of mutualism. Now cuscuta growing on the hedge plant. Cuscuta is a plant that does not have the chlorophyll pigments, so it cannot obviously do photosynthesis. So it depends on the normal plants for food and everything. So it has, you know, cuscuta, which is yellow in color, you might have seen it. It slowly develops some projections into the plant's phloem and xylem tissue, and it takes in all the nutrients up there. So this is an example of parasitism. This is an example of parasitism. This came in this model question paper. So we make sure you know the answers of all these things. Okay. Now, predators are prudent and conduits in nature. Support your answer by giving reason. What do you mean by prudent? So, predators are not like, I am going to eat all the prey. So, I am a tiger, I am not going to eat all the prey. Because if I do so, will I have the food next day? No. So, basically, I am going to target the weaker ones, and I am going to make the stronger ones there only. Because obviously, the weaker ones, it's easy to target them, to eat them. And also, if the stronger ones are there, they are going to reproduce. Okay, the weaker ones, what reproduction they will be doing? The stronger ones, they will be reproducing, and you can get the kids, I can eat them. You got it? So they are prudent, they think about it. If they keep on eating, my dear children, don't you think the whole ecosystem stability will be lost? Yes, it will be completely lost. If I vanish away all the deers of one trophic level, my dear children. Now, obviously I don't have any food. The plants will also flourish. Okay, the plants will also flourish. I won't have any food. I will die. Get it? Now they are conduits in nature. What do you mean by conduits? Basically, conduits as in they are like connecting link in the nature. Through them only the energy is being passed. So predators are the ones. For example, I have plants, and the plants are being eaten up by a rabbit, and the rabbit is being eaten up by a tiger. So isn't it a conduit, because it is passing down the energy? It is passing down the energy with the help of it. So which is why it is known as conduit. Now this tiger can be eaten up by an eagle or falcon or whatever is there. You get it? Okay, basically a falcon, anything that you can put, a vulture, not eagle, basically I can put vulture. I'm talking about dead tiger, okay. Got it? So how do you frame this answer? So predators plays an essential role in maintaining ecological balance by regulating prey population. They are considered prudent because overexploitation of prey would lead to their own decline due to food scarcity. Okay, overexploitation cannot be done, because in that way they are going to have food scarcity, right? Predators are conduits in the energy flow of an ecosystem, transferring energy from prey species to themselves and other trophic levels. For example, lion hunting herbivores ensure weak and diseased individuals are removed, which indirectly improves the prey population health as well. Yeah, that is also one more very important thing. Now, to lessen the impact of predation, prey species have evolved different defensive methods in plants and animals. Support your answer by giving one example for plant and an animal. So over here, let's see. So in animals, some species, you know, of insects and plants, they are cryptically colored, they camouflage, okay, so that they are not visible. Okay, that is one thing. So some are poisonous and therefore avoided by the predators. Obviously, if once I eat them, I won't come and eat them again. Now, monarch butterfly is highly distasteful to its predators due to the presence of special chemical in its body. So in its larval stage, it feeds on some plant which has certain very bad chemicals, my dear children. So it becomes highly distasteful even in adult stage. Now the chemical is acquired by feeding on the poisonous weed during caterpillar stage. So monarch butterfly, all the insects and the frogs, you can give example for that, and some poisonous animals, you can give example for that. In the case of plants, you have what thorns in acacia, cactus are morphological means of defense. Yes, they are structural means of defense, my dear children. Apart from that, many plants produce and store some chemicals which make the herbivore sick when they are eaten, inhibit feeding, digestion and disrupt reproduction, or even kill the predators. They even kill the predators, my dear children, because they are storing certain very important toxic substances. Now, calotropis produces poisonous cardiac glycosides against herbivores. So if herbivores comes and eats it, obviously they will get diseased. So calotropis is an example example, thorns in acacia and cactus is an example, and apart from that, the chemicals which are being stored in them, that is also an example. So you can write all these examples. You are asked to 1-1, you can either write 1-1. So the best one will be monarch butterfly from here, and calotropis and thorns in acacia would be the best one from here. Done? Chalo. Next, explain mutualism with examples. How can you explain mutualism? Mutualism is when both of them are benefited. Both of the organisms are benefited. So if we talk about lichens, lichen is an association of what, children? Algae and fungi. Algae and fungi. Algae will be providing the fungi with nutrients, the compounds, because it can do photosynthesis, okay, right? It can do photosynthesis, provide nutrients. What about fungi? Fungi will be providing mineral nutrients by seeping things. So it is providing food, and the fungi will be providing nutrients, because fungi can get deep into the things and take in the nutrients. You get it? So that is lichen, my dear children. Now you have mycorrhizae. Mycorrhizae is basically again a symbiotic association of plants and fungi. The plants can give food to them, and the fungi will provide what? Nutrition such as phosphorus from the soil. So that is again an example. Apart from that, the fig and the wasp, okay, that is an example. So lichens present close relationship between a fungus and an algae or cyanobacteria. The fungi help the algae in absorption of essential nutrients from the soil, whereas the algae provide food for the fungi. Similarly, between fungi and roots of higher plants, that is mycorrhiza. Okay, fungi help the plant in absorption of essential nutrients from the soil, while the plant provides the fungi with carbohydrates. Now plants and pollinating agents, yeah, plant provide nectar and pollen for the pollinating agents, and also space, and animals help as agents, what? Agent of pollination. Now plants and seed dispersing agents. Animal disperse the seed, same thing, you can say that. And particular species of fig tree is pollinated by species of wasp only. The wasp pollinates the fig inflorescence while searching for suitable sites for oviposition, that is for laying egg. In turn, basically, the fig provides the wasp some of its developing seed as food for the developing wasp embryo. So that is also there. So both of them are helping each other. You can give all these examples, like in mycorrhiza, plants and pollinating agents, and also the wasp thing. Okay. So what is commensalism, and explain with any two example, and you have to state Gauss's competitive exclusion principle. So my dear children, commensalism is when one organism is getting the benefit, another organism is neither harmed or nor is getting the benefit. Examples are orchids growing as an epiphyte on the mango branch. Okay, that is one example. Basically, the orchid is staying up there because it wants sunlight and everything. Is it harming the mango plant? No. Second thing, clownfish living among the tentacles of sea anemone. Okay, so clownfish, when it lives among the tentacles of sea anemone, my dear children, this is an example. Why? Because the sea anemone is providing protection to the clownfish is not. Now, barnacles on the back of whales, that is one thing. Now, cattle egret and the grazing cattle. So, grazing cattle, when it moves around, what is happening is that cattle egret will be feeding on all the small things that are going to come off. Okay, so grazing cattle are not going to get any benefit, which is why it is coming under commensalism. And with that, I want to correct one very important thing that is up over here. That is here. So sea anemone and clownfish, it is not mutualism. Actually, they have certain mutual relationship. Obviously, they benefit each other. Okay, but according to your NCERT, it is commensalism. It is commensalism. So please make sure you are doing this correction. Okay, not mutualism but commensalism. Now going ahead, where were we? Yeah, now we are talking about Gauss's competitive exclusion principle. It states that if basically there are two organisms or more than two organisms, close related species, if they are in an area and basically they are utilizing same resources, then they cannot coexist with each other. The competitively inferior one will be obviously eliminated slowly or gradually, and the competent and dominant one will be staying over there. So two closely related species competing for same resources cannot coexist indefinitely, and the competitively inferior one will be eliminated eventually if the resources are limiting, my dear children. So that is Gauss's competitive exclusion principle. Got it? Now next, what are ectoparasite and endoparasite? Ectoparasite is the one that will be staying outside the body, for example lice, ticks and things like that. Endoparasite is the one that stays inside the body of the host organism. For example, you have tapeworm and things like that. So how do you frame it? Parasites feed on the external surface of host organism. Parasites that live inside the host organism at different sites are what? Basically live inside This is ectoparasite and this is endoparasite. Now list any 3 parasitic adaptation in animals. If they are parasite inside the body, what are the adaptation? Loss of unnecessary sense organs, obviously they do not want that. Presence of adhesive organs so that they can remain stuck. Okay, loss of digestive system, and then high reproductive capacity is also there. Apart from that, complex life cycle is also involved in them. So these are the features. Write any 4 factors that influence population density, my dear children. So you have natality, you have mortality, you have immigration and emigration. Natality as in the birth rate, mortality as in death rate, immigration as a number of organisms that is coming in, emigration as in who is leaving the population. Obviously this will influence the population density. Now you have to draw a graph showing exponential growth curve. What do you mean by exponential growth curve? Here they are still growing. There is no limiting factor. They are just growing. So how does the graph appear? Slowly it will take some time to grow and then it will be shooting up. There is no limiting factor, which is why it is known as exponential growth curve. Okay, it is represented by d and by dt is equals to Rn, where R is intrinsic rate of natural increase and N is the population size. Okay, this is population density, this is time and everything. Work? Cool. Now, what is population growth? Explain factors responsible for fluctuating the population density. So, what is population growth? The size of the population changes depending on the availability of food, predation pressure and reduced weather. Okay, and weather. So, population size fluctuated due to changes in 4 processes, 2 of which is natality and immigration, and contribute to increase in population density, these 2, okay, and 2 of them basically decrease the population density, that is mortality and emigration. So, you have to mention all this thing. What is population growth? Population growth is the growth in the size of the population at a given time, at a given area, that is population growth. Okay, and then these are the factors which affects the population growth or the density, that is natality, mortality, immigration and emigration. So, natality and immigration will have a positive effect because obviously you are increasing the population size, and over here mortality and emigration will have a negative effect because you are decreasing the population size. Got it? So, I am so sorry I am going really fast because the time is going really up. Okay, so but I am hoping I am covering everything up. So yes, ecosystem has no 5-marker question, my dear children. Even biodiversity and conservation, it is out of 4 marks. So over here this is a 4 and this is also a 4, which is why there are no 5-marker questions. And with this we are completing all the 5-marker, all the important 5-marker questions of all the chapters, my dear children. This video is really important for you. I'm so sorry if at all at some point I have done some mistake, okay, so please forgive me. I would be correcting it. If at all it's there, if at all it's not there, then there is no point of correction. But yes, mostly it is all fine, but it's a long video, so yes, so that is there. And thank you for watching. Make sure you're writing the answers in points. It's 5-marker, so obviously you have to write the answer in points. If you're not doing so, you might lose up your marks. So don't do that mistakes. So I hope that was helpful. Not forget to like, share and subscribe to our channel, and do let me know if it was helpful for you, and please ask me doubts in your doubt section in the comments. Okay, so bye bye, take care.